Somewhere in the world right now, roughly 1 in every 14 people has blood that gets flagged differently. Not dangerous, not diseased, just different. A small protein marker that most humans carry is simply absent, and that absence has been enough to build an entire mythology around. Secret ancestry, ancient alien origin, superhuman resistance to disease. A bloodline that traces back not to Africa or the Middle East, or any place on a normal map, but somewhere else entirely. Somewhere that doesn't exist in the history books, or maybe never existed at all. We're talking about Rh-negative blood. And before we go anywhere near the myths, which we absolutely will, let's just sit with the fact that this one small biological detail has generated more internet conspiracy theories, more breathless YouTube speculation, more late-night rabbit holes than almost any other quirk of human genetics. That's interesting by itself. That's worth asking about. Why does a blood type carry so much cultural weight? If you're here to fall asleep to something genuinely worth thinking about, that's exactly what this is for. So get comfortable, and before you drift off, drop a comment and tell me where you're listening from and what time it is there. I check them and it never gets old. It's midnight in Melbourne, it's Tuesday morning in Dublin, it's someone on a train in Tokyo at 6 a.m. That's something. So let me know. Now, back to the blood. The story of Rh-negative touches something most history channels don't think to connect. A single scientific discovery made during World War II, the deaths of thousands of infants across centuries that nobody could explain, an isolated mountain population in Europe whose language has no known relatives anywhere on Earth, and a very human tendency to look at something rare and strange and ask, what does this mean about me? What we're going to do tonight is follow all of it. The actual science, which is genuinely fascinating and does not need to be exaggerated. The history of blood research, which is wilder than most people realize and involves a lot of early medical disasters before it involves any triumphs. The geography of where Rh-negative clusters appear around the world and what migration patterns might explain that. And then the mythology. The ancient astronaut theories, the Atlantis bloodline stories, the Nephilim descendants, the Starseed community, all of it examined not to mock, but to understand why these stories exist and what they're actually doing for the people who find meaning in them. Because that's the more interesting question, isn't it? Not just whether the claims are true, but why they emerged, why they spread, and why a blood type became the center of an identity. The core question underneath all of it is deceptively simple. What is Rh-negative, really? Not in the mythology version, not in the conspiracy version, but in the actual biological, historical, evolutionary sense. And the honest answer is that even scientists don't have a complete picture. There are genuinely open questions about why the Rh-negative variant persists in human populations at the frequencies it does. There are real debates about ancient migration and selection pressure that don't have settled answers yet. The mystery isn't invented, it's just not the mystery most people are telling. Here's a few things you can look forward to as this unfolds. In the early 20th century, doctors were performing blood transfusions that were killing people, and they couldn't figure out why. The discovery of the Rh factor came out of research on rhesus monkeys, and it solved a puzzle that had been haunting obstetricians for generations. Mothers and their own unborn children, sharing a womb, sometimes sharing an immune response that could be fatal. The science that explained that changed medicine permanently, and also put Rh-negative on the map in a way it had never been before. And then, decades later, something else happened. The information moved from hospitals into culture, and culture does what culture always does with something rare and unexplained. It reaches for meaning, it builds stories, it finds patterns that may or may not be there. We'll sort through all of that carefully. No definitive claims where the evidence doesn't support them. No debunking for its own sake either. Just the history, the science and the stories laid out slowly, in order the way a long night deserves. Rh-negative blood has been casting a shadow across medicine, mythology, and human identity for longer than most people realise. Let's go back and find out where that shadow started. Start with the simplest possible version of what's actually happening here, because the mythology around Rh-negative blood tends to make it sound far more exotic than the underlying biology is. And if you understand the biology first, the history makes more sense, the mythology makes more sense, and honestly, the genuine mystery becomes a lot clearer too. Your blood is classified in multiple ways at the same time. The system most people know is ABO, which gives you your AB or O designation. That system is based on specific molecules called antigens that sit on the surface of your red blood cells. Think of antigens like small molecular flags. Your immune system learns early in your development which flags are yours, and it treats anything with different flags as foreign. That distinction, self versus not self, is the cornerstone of how your immune system works. Abotyping is essentially cataloguing which flags you're flying. The Rh system is a separate layer of the same basic idea. It's actually a much larger and more complex system than ABO when you look at it closely, involving dozens of different antigens. But for almost all practical purposes in medicine and in everyday conversation, when someone says Rh positive or Rh negative, they're talking about one specific antigen. It's called the D antigen. And the entire positive-negative shorthand comes down to this. Do you have it, or do you not? If your red blood cells carry the D antigen, you're Rh positive. If they don't, you're Rh negative. That's it. Rh positive and Rh negative aren't descriptions of blood quality or blood strength or some hierarchy of biological status. They're just a way of noting the presence or absence of a single protein on the surface of a cell. Now, here's where it's worth pausing, because this is where the mythology almost always slips in unnoticed. The absence of the D antigen is not a disease. It is not a deficiency. It is not evidence of a mutation in the catastrophic sense of that word. Rh-negative people have perfectly functional red blood cells. Their blood does everything blood is supposed to do. The only time the Rh factor becomes medically significant is in specific contexts involving mixing, specifically transfusions and pregnancy, and we'll get deeply into both of those later. Outside of those contexts, Rh-negative blood is simply a variation, a flag you're not flying, nothing more. It's also not evidence of a separate species. This point needs to be made plainly because that claim circulates widely. There are no two groups of humans with blood types so different that they represent distinct species. Homo sapiens is one species. Our blood types are variations within that single species, like variations in eye colour or height or earwax consistency. The fact that some populations have different frequencies of a particular blood type tells us something interesting about genetics and migration, and possibly about selection pressures over time. It doesn't tell us that one group descended from extraterrestrials and another didn't. So where does Rh-negative come from genetically? Here's the simplified version. The Rh-positive trait, meaning the presence of the D antigen, is inherited through a gene that produces that antigen. Rh-negative individuals typically have a version of that gene or a deletion in that region of their DNA that doesn't produce the D antigen. The details are more complex than that, and scientists are still working out some of the finer points, but the core of it is that Rh-negative is a recessive trait. To be Rh-negative, you generally need to inherit the relevant variant from both parents. If you inherit one copy of the gene that produces the D antigen and one copy that doesn't, you'll be Rh positive, but you'll be carrying a hidden copy that you can pass on. This is why family patterns can genuinely surprise people. Two Rh positive parents can have an Rh negative child. That's not a medical anomaly or a sign of something unexplained in the family tree. It's just recessive inheritance doing what recessive inheritance does. The same basic math applies to dozens of traits across the human genome. But because most people don't think carefully about genetics until something unexpected surfaces, the Rh-negative child of two Rh-positive parents can feel like a mystery. And when people encounter something that feels like a mystery in their own biology, they often go looking for explanations in all directions at once, some scientific and some very much not. The question of rarity is another place where careful thinking matters, because Rh-negative isn't equally rare everywhere. Global averages can be deeply misleading. Across the entire human population, Rh-negative individuals make up roughly 15 to 17% of people. That's not vanishingly rare. It's actually fairly common as genetic variants go. But those numbers are heavily shaped by which populations are included in the calculation. In parts of West Africa, Rh-negative frequencies drop to around 1 to 3%. In many East Asian populations, the number is even lower, sometimes under 1%. In some indigenous American populations, it's extremely rare as well. But in Europe, and especially among certain populations in southern France and northern Spain, the frequency climbs dramatically. In some Basque communities, estimates have placed the Rh-negative frequency as high as 30 to 40%, which is extraordinary. More than 1 in 3 people in a small, geographically isolated population carrying a trait that in much of the world barely shows up at all. That geographic clustering is real, and it's one of the things that has genuinely captured scientists' attention. We'll spend a lot of time later trying to understand what those patterns might mean about ancient migrations and population history. For now, the important thing is simply to recognize that when someone says Rh-negative is rare, the honest answer is, it depends entirely on where you're standing. And that variation, those dramatic differences between populations in different parts of the world, is part of what makes this trait so narratively attractive. Human beings are very good at noticing patterns and then reaching immediately for explanations. When you see a trait that clusters strongly in one region, that spreads unevenly across the world in ways that don't follow the distribution of other genetic markers, that seems to increase in isolated populations and decrease in large, interconnected ones. Your brain starts asking why. That's a reasonable instinct. Scientists ask the same question. The difference is in where the search for answers goes. The mythology around Rh-negative blood didn't emerge from nowhere. It emerged from real data that genuinely needed explaining. The clustering is real. The geographic patterns are real. The questions about why this particular variant persists at the frequencies it does, and why selection pressure hasn't changed those numbers more dramatically over time, are real questions without fully satisfying answers yet. That gap between what we observe and what we can explain with certainty is exactly the space where storytelling thrives. It always has been. Ancient aliens, lost civilizations, hybrid bloodlines, they all moved into territory that conventional science hadn't fully mapped. Understanding the actual biology doesn't make the story less interesting. It makes it more interesting because now you know what the real questions are. Not whether Rh-negative people are secretly descended from Pleiadian-style beings, but why this variant exists in human populations at all, why it clusters the way it does, and what the history of those populations can tell us about where we all came from. Those questions have actual, complicated, genuinely unresolved answers. And to understand why anyone was asking them in the first place, we have to go back to a time when doctors had almost no idea how blood worked at all, and were learning the hard way. When doctors first started moving blood from one person to another, they were doing something astonishing with almost no map. They knew people could bleed to death. They knew that blood seemed to be life itself. They had stories from animals, from myths, from earlier medical traditions. But they didn't know that inside that dark red fluid were invisible differences that could mean the line between survival and catastrophe. Early transfusions were, in many ways, experiments performed on the edge of ignorance. In the 17th century, Physicians in Europe tried transferring blood between animals, and then from animals into humans, a lamb into a man, a calf into a fevered patient. Sometimes the person would survive and even seem to improve. Other times they would develop chills, fever, pain, dark urine, trouble breathing, and then simply die. The pattern was terrifyingly unpredictable. So the stories went like this. Transfusion is dangerous, but occasionally miraculous. One patient survives and is hailed as proof the method works. Another dies, and the entire practice is denounced as reckless, immoral, or even blasphemous. Laws were passed, experiments were banned. The idea of transfusion retreated into the shadows for decades at a time. What nobody could see was that the blood itself was not a single uniform substance. On the surface of every red blood cell, those tiny molecular flags were already deciding the outcome. But because the flags were invisible, the line between help and harm felt essentially random. By the 19th century, medicine had moved into a more systematic phase. Hospitals were formalizing, surgery was becoming more common, especially in cities. Anesthesia made it possible to perform longer, more invasive operations. But with every improvement came a cost, more chances to lose blood and more desperate attempts to replace it. Again, doctors tried transfusion. This time, they were often moving blood directly from one human to another instead of from animals. And again, the pattern repeated. Some patients improved. Others developed what we now recognize as acute hemolytic reactions, their bodies attacking the transfused blood as foreign, destroying red cells, releasing toxins, overwhelming the kidneys, the heart, the lungs. They didn't have that language yet. They just had the image of a patient who was already weak, suddenly shaking with chills, spiking a fever, their urine turning red or brown, their pulse failing. To the doctors at the bedside, it felt like Russian roulette. The same procedure, apparently the same method, but radically different outcomes. When a technique behaves like that, people look for the wrong kinds of patterns. Maybe it depends on the donor's age. Maybe it depends on how long the blood sits in the vessel. Maybe it depends on whether the donor is strong or weak, pure or tainted. In the absence of actual mechanisms, medicine fills in with folk theories. This is one reason the discovery of the ABO system hit like a quiet earthquake. In 1900 and 1901, the Austrian physician Karl Landsteiner was working in Vienna, doing something simple and methodical. He took samples of blood from different people. He separated the serum from the red blood cells. Then he mixed them, one person's serum with another person's cells, cataloguing what happened. Sometimes the red cells stayed floating calmly in the fluid. Sometimes they clumped together, a process called agglutination. Sometimes they clumped and then broke open. The patterns weren't random. Certain samples consistently reacted with each other and not with others. From this, Landsteiner inferred that there must be distinct types of blood, defined by the presence or absence of specific antigens. He labelled them A, B, and what he called C, which would later be renamed O. Shortly after, other researchers added AB, the type that carried both A and B antigens on its red cells. For the first time, the invisible flags had names. This changed transfusion from a gamble into a procedure that could be planned. If you knew a patient's ABO type and the donor's ABO type, you could predict whether their blood would coexist peacefully or attack each other. You could map compatibility, you could learn through trial and error, and eventually large datasets that O blood could be given more broadly, that AB individuals could receive more broadly. War surgery felt this impact very quickly. The First World War turned European battlefields into vast anatomy laboratories, in the bleakest possible sense. Men bled out from shrapnel wounds, from amputations, from infections. For the first time, armies tried to scale up transfusions systematically. They began testing soldiers, keeping track of their blood types, matching donors to recipients. The death rate from shock and hemorrhage dropped. Battlefields became places where blood typing wasn't just a lab curiosity, it was a matter of logistics. This is something classification systems do over and over in human history. Once you can sort something into named boxes, it begins to feel more real, more solid, more like an identity. Being type A or type O started as a lab code, a practical note on a chart. But the idea that your blood had a type carried an almost immediate psychological weight. People were already used to categories of race, nationality, religion. Now medicine was adding a quiet new layer, invisible groupings written in your cells. In some cultures, blood type would later be linked, very loosely and unscientifically, to personality. In others, it fed into fantasies about pure and impure bloodlines. The science was about antigen-antibody reactions. The imagination went much further, as human imagination tends to do. But even after ABO was known, the mystery of the occasional lethal transfusion didn't disappear entirely. There were still cases where everything seemed to match on the ABO chart, and yet the recipient reacted violently. Fevers, jaundice, anemia, stillbirths. Something else was happening beneath the named categories. For several decades, that something else remained effectively invisible. Part of the reason was that the ABO system solved most of the worst, most dramatic transfusion disasters. If you stopped putting a blood into B recipients and vice versa, your immediate catastrophic reaction rate dropped dramatically. When an intervention works that well, the residual problems can be easy to overlook or to explain away as infection, bad technique, or individual fragility. Another part of the reason was technical. Detecting more subtle antigen systems like Rh required more precise reagents, more controlled experiments, and a willingness to believe that the surface of the red blood cell might carry not just 2 or 3 kinds of flags, but dozens of them. That's a conceptual leap once you have a classification system. It's tempting to think it's complete. So, for a while, medicine lived in a kind of partial clarity. ABO typing was standardizing. Hospitals in industrialized nations began to build blood banks. Military planners integrated blood supply into their strategies. Donor drives, storage protocols, typed tags on soldiers' dog tags. Blood was becoming not just a fluid, but an organized resource, catalogued and controlled. Behind that practical organization was a particular social world. Large centralized hospitals were replacing smaller local healers. Medical authority was becoming professionalized, credentialed, increasingly tied to the state. Governments, especially in times of war, saw blood as a strategic material. If you could keep your wounded alive and return them to battle or at least home, you preserved not just lives but manpower, morale, and political stability. Standardization fit that world perfectly. Typing systems, labels, protocols. Who can donate to whom? How much can be stored? How long it remains viable? Blood became something you could regulate, legislate, and plan around. In that environment, a new blood group system was almost bound to be discovered. There were too many outliers. Babies dying shortly after birth despite seemingly careful care. Women who had one healthy child and then, mysteriously, repeated pregnancy losses. Transfusion reactions in abo-compatible pairs. The data didn't quite align with the existing boxes. And when a classification system stops explaining the edge cases, scientists start looking for a deeper layer. That deeper layer is where the Rh factor lives. But it wouldn't enter the historical record until a particular set of experiments, on a particular group of animals, in a particular lab, finally revealed that there was another antigen system sitting quietly on the surface of red blood cells, shaping life and death without a name. That's where we're going next. Into the moment when Rh stopped being invisible and became a problem medicine could finally see and try to solve. The story usually goes like this. One day, scientists discovered a new blood factor in monkeys, named it RH, and suddenly everything made sense. The reality was slower, stranger, and much more human. By the 1930s, the ABO system was an accepted part of hospital life in Europe and North America. Doctors ordered type and cross matches the way they ordered X-rays and morphine. Laboratory technicians spun blood in centrifuges, labelled glass tubes with careful handwriting, and checked charts before transfusions. Most of the time, this worked. When you kept A away from B, B away from A, and handled O and AB appropriately, the dramatic, immediate transfusion disasters mostly stopped. But not entirely. There were still the mystery cases. A patient would be carefully typed and matched. Their donor would be ABO compatible. The transfusion would begin. For a while, everything seemed fine. Then, sometimes hours later, the patient would develop a fever, or their red cells would begin to disappear from circulation, or their urine would darken, or they would become jaundiced. Sometimes they survived, sometimes they didn't. Pathologists performing autopsies could see the damage. Broken red cells, overloaded kidneys, signs of a body that had turned against the very blood meant to save it. And yet, on paper, the transfusion had followed all the rules. In another corner of medicine, a different puzzle was quietly accumulating. Obstetricians and midwives before them had long known a cruel pattern that didn't fit neatly into any theory. A woman might have her first baby without complications. Then, with her second or third pregnancy, tragedy would begin to strike. The fetus might die in the womb late in pregnancy, or the baby would be born swollen, anemic, with severe jaundice, sometimes with fluid filling the abdomen, chest, and under the skin. This condition would eventually be called erythroblastosis fetalis, or hemolytic disease of the newborn. At the time, it was just another heartbreak with no clear cause. Mothers blamed themselves. Doctors blamed infections, bad luck, weakness. The pattern, first baby fine, later babies at higher risk, was noticed but not understood. There was no obvious infection, no consistent environmental toxin, nothing easy to point to. These two mysteries, delayed hemolytic transfusion reactions and recurrent newborn illness, did not at first look like the same problem. They lived in different specialties, with different practitioners, different journals, different conferences. The physician who fought to stop a postpartum hemorrhage with transfused blood was not necessarily the same physician who watched a jaundiced newborn fade. For the RH story to begin, those worlds had to come closer to the laboratory bench. In New York, in the late 1930s, Karl Landsteiner, the same Landsteiner who had defined the ABO system decades earlier, was still working. By this point, he was at the Rockefeller Institute. Older, more established, but still engaged in the same basic act, taking small samples of blood, mixing them in different combinations, watching for patterns. He and his colleague Alexander Wiener were interested in the idea that there might be additional antigens, additional flags on red blood cells beyond A and B. The hint was simple: if some transfusion reactions still happened despite ABO compatibility, something else must be at play. To study this, Landsteiner and Wiener turned to animals, specifically rhesus macaque monkeys. They immunized rabbits and guinea pigs with red blood cells from these monkeys. The animals' immune systems did what immune systems do. They produced antibodies against whatever they recognized as foreign on the monkey cells. Then came the critical question: would those antibodies react with human red blood cells? The answer, for some human samples, was yes. When the anti-rhesus serum was mixed with red cells from certain people, those red cells clumped. When it was mixed with red cells from other people, nothing happened. There it was again, a pattern. Some humans seemed to carry a particular antigen on their red cells that was similar enough to a monkey antigen that the same antibodies reacted to both. Others did not. Landsteiner and Wiener called this new specific reactivity the Rasch factor. after the rhesus monkeys that had made the experiment possible. This is where the story usually gets compressed. Textbooks will say, the Rh factor was discovered in 1940 by Landsteiner and Wiener in experiments with rhesus monkeys. That's not wrong, but it leaves out just how much confusion was baked into that first naming. For one thing, the antigens on human red cells that really matter clinically in the Rh system are not actually identical to the original monkey antigen that gave the factor its name. Later work would show that the anti-rhesus antibody Landsteiner and Wiener studied wasn't targeting exactly the same structure as the one involved in most human transfusion and pregnancy problems. The word Rh stuck anyway. This happens often in science. A first name is convenient, memorable, and tied to an experiment that opened the door. By the time the details become more nuanced, the name has already circulated through papers, conferences, hospital protocols, and then into general medical speech. So we now talk about Rh factor as if it were one simple thing. But in reality, the Rh blood group system is a cluster of closely related antigens. The most important one for transfusion and pregnancy is called D. When you hear someone say Rh positive, what they really mean is D antigen present. When they say Rh negative, they mean D antigen absent. That distinction, D present or D absent, would become the axis on which thousands of lives would quietly turn. At first, though, the situation was a mess. Different researchers were using slightly different sera, different immune cocktails to test for rhesus-like reactivity. Some of these sera recognized the D antigen, others recognized related antigens. Some labs reported that a patient was Rh positive, while others flagged the same person as Rh negative, because they were actually looking at different molecular targets. There were arguments in journals, letters back and forth, attempts to standardize reagents. Slowly, through the 1940s, a kind of consensus emerged. The antibody that caused the most severe clinical reactions in humans was the one directed against what we now call D. Once that was clear, the picture changed. The mysterious transfusion reactions in ABO-compatible pairs began to make more sense. If a person who lacked the D antigen, someone who was Rh negative, was exposed to Rh positive blood, their immune system could slowly become sensitized. The first exposure might not cause much trouble because it takes time to build a strong antibody response. But later exposures could provoke a powerful reaction. The immune system, once trained to recognize D as foreign, would attack any incoming D-positive red cells with intensity. Now look back at those obstetric puzzles. A mother who is Rh negative carrying an Rh positive fetus is, in a quiet, immunological sense, carrying something foreign. During the first pregnancy, the mother's immune system often doesn't get enough fetal blood exposure to fully learn the D antigen. The baby is born healthy, but childbirth itself can mix maternal and fetal blood. So can miscarriage, abortion, certain kinds of trauma, or invasive obstetric procedures. Those events can sensitize the mother. Her immune system begins to produce anti-D antibodies. In a later pregnancy with another Rh-positive fetus, those maternal antibodies can cross the placenta, enter the fetal circulation, and begin to destroy the baby's red blood cells. The result looks exactly like erythroblastosis fetalis. The parallel between transfusion reactions and hemolytic disease of the newborn is obvious in hindsight. Both are cases of an Rh-negative immune system attacking Rh-positive red cells. But for the clinicians living through the 1930s and early 1940s, connecting these dots required a leap to see pregnancy not just as a biological state, but as a kind of prolonged, subtle transplant, to see newborn illness not just as bad luck, but as the echo of an earlier immunological event. The leap was made piece by piece. In 1939, Philip Levine and Rufus Stetson in New York described a woman who had a stillborn baby with severe hemolytic disease. She herself then had a transfusion from her husband, who was abo-compatible. Despite that, she had a severe hemolytic reaction. Levine showed that her serum reacted strongly with her husband's red cells, and with the red cells of about 80% of people they tested, but not with others. Something in her immune system was attacking a common antigen that she lacked. This was before Landsteiner's rhesus experiments were fully known, so Levine didn't call it RIR, but they were looking at the same phenomenon from the obstetric side. When the rhesus-based Rh discovery and the clinical observations about newborn disease met, the mystery began to resolve. The same basic incompatibility was playing out in transfusion rooms and in delivery wards. Even then, the clarity reached different places at different times. Major research hospitals in the United States and Europe started adopting Rh typing in the early 1940s. Blood banks layered this new classification on top of ABO. Charts and donor cards acquired another little symbol, a plus or a minus. Nurses and technicians learned new routines. Rh-negative patients were flagged for special care. But knowledge doesn't spread evenly. Smaller hospitals, rural clinics and colonized regions often lagged behind. Reagents for Rh testing had to be produced, standardized, shipped, stored. Training had to occur. Local authorities had to decide that this new test was worth the cost. For a period of years, a person's risk of Rh-related complications depended not just on their genes, but on where they lived, which hospital they reached, which doctor happened to be on call, whether that doctor had recently read the right journal or attended the right meeting. This is how many medical breakthroughs move through the world, not like a light switch flipping from off to on, but like the slow spread of dawn, uneven across landscapes, leaving pockets of darkness for longer than we like to remember. Still, by the mid-20th century, the basic framework was in place. There was such a thing as Rh. It came in a positive and a negative form, at least at the level of the D antigen. It explained certain transfusion reactions that abo could not. It explained why some mothers lost multiple babies despite seemingly healthy pregnancies. The next question was brutal and practical. If you can finally see the mechanism behind so much hidden loss, can you stop it? Answering that question would lead to one of the most consequential interventions in modern obstetrics, and to the moment when Rh status stopped being a purely laboratory curiosity and became, for many families, the difference between grief and survival. To understand what came next, it helps to slow the story down and look at it from inside a single body. Imagine an Rh-negative woman on her red blood cells. The D antigen simply isn't there. To her immune system, that blank space is normal. It's home. Now imagine she conceives a child with an Rh-positive partner. The fetus inherits the D antigen. Those tiny developing red blood cells circulate quietly inside the womb, doing their job. For most of the pregnancy, mother's blood and baby's blood don't mix very much. They are close but separated. Two systems exchanging nutrients and gases through the placenta, without usually swapping many whole cells. From the outside, everything can look perfect. Routine checkups. A growing belly, heartbeat on the examiner's stethoscope. But at the boundary between those two circulations, a kind of secret handshake is being set up. If a little bit of fetal blood crosses into the mother's circulation during late pregnancy, or a small bleed, or especially during delivery, her immune system can suddenly see something it has never seen before. Red cells carrying the D antigen, and the immune system, being what it is, takes notes. The first time, it is usually slow. Antibody production ramps up gradually, experimenting, learning the shape of this new target. The pregnancy may end with a healthy, Rh-positive baby in the mother's arms. On the surface, nothing seems wrong, but inside, the blueprints have been filed away. B cells carrying the memory of D as foreign now exist. The woman has become sensitized. In a later pregnancy with another Rh-positive fetus, the timing is different. Those memory cells don't need to learn from scratch. They are ready. They can produce anti-D antibodies quickly and in large amounts. Those antibodies, small and mobile, cross the placenta with ease, move into the fetal circulation, and begin to bind to the baby's red blood cells. The baby's own immune system isn't the aggressor. The attack is coming from the outside, from the parent whose body is also feeding and sheltering it. This is Rh incompatibility in its most dangerous form, an Rh-negative mother with circulating anti-D antibodies, and an Rh-positive fetus whose red cells carry the very antigen those antibodies are trained to destroy. Once the antibodies bind, fetal red cells are marked for destruction, mainly in the spleen and liver. The baby becomes progressively anemic. To compensate, the tiny bone marrow, and even organs that made blood only in early development, like the liver, start pushing out immature red cells as fast as they can. Under the microscope, you see these immature forms crowding the blood. That's where the older name erythroblastosis fetalis comes from, a fetus full of young, rushed-out red cell precursors. As the anemia worsens, the fetal heart works harder and harder, trying to keep oxygen delivery adequate, with fewer functioning red cells. Fluid begins to leak into tissues and body cavities. The liver and spleen swell. Severe cases develop a condition called hydrops fetalis, where the fetus becomes profoundly swollen with fluid. Even if the baby makes it to birth, the trouble isn't over. The breakdown of all those red blood cells releases large amounts of bilirubin, a yellow pigment. The newborn becomes deeply jaundiced. At high enough levels, bilirubin can cross into the brain and cause permanent damage, kernicterus, a form of brain injury that can leave survivors with movement disorders and hearing loss. Today, with modern neonatal care, exchange transfusions, phototherapy, and aggressive monitoring, we can often intervene if we see this process starting. For most of human history, none of that existed. All anyone saw was the surface, a pattern of loss. A first pregnancy often fine, a second sometimes with late miscarriage or a stillbirth, a third worse, a family in which nearly every male child seemed to die in the womb, a woman whose sisters and mother also had bad luck with babies, a line of graves that seemed to follow certain marriages. Midwives noticed, they might say quietly, "Her babies don't live," in the same tone used to describe a family tendency toward bleeding or madness. Physicians in the late 19th and early 20th centuries wrote case reports describing familial hydrops, icterus gravis neonatorum, severe jaundice of the newborn, or families where multiple siblings died with swollen bodies and yellow skin. They had words for the appearance. They did not have a mechanism. Some blamed infections, though no consistent germ was found. Some invoked vague ideas of maternal toxicity or constitution. Others reached for moral or spiritual explanations. In many cultures, a woman who lost multiple infants was pitied or quietly suspected or encouraged to accept that God did not mean her to have children. Grief folded into shame. Husbands and wives blamed themselves or each other. In communities with limited medical language, the pattern was seen as a curse or a sign that this particular marriage was wrong in some ineffable way. Even after obstetricians could describe hemolytic disease of the newborn as a recognizable syndrome, anemia, jaundice, enlargement of liver and spleen, fluid accumulation, the cause remained opaque. It was as if certain wombs had become hostile environments for certain babies, for reasons no one could clearly state. Looked at from a distance across hospital archives and parish records, a chilling regularity emerges. Some families lost baby after baby in the 1920s and 1930s in exactly the pattern you would now predict from Rh incompatibility. The firstborn living, the second jaundiced and sick, the third dying in utero, the fourth only surviving with desperate measures, if at all. The cost was not just emotional. In societies where children were economic security, future labour and old age support, repeated infant loss could destabilise entire households. Women with a history of bad pregnancies might be marginalised or pressured into dangerous remedies. Some simply stopped trying. Others kept going, hoping this time would be different, walking again and again into the same invisible immunological trap. When the Ulrich story began to crystallise in the 1940s, it offered for the first time a coherent explanation that did not blame the mother's character or inscrutable fate. It said quietly but firmly, "This is a specific mismatch of blood markers. This is a mechanism. And mechanisms, unlike curses, can sometimes be interrupted." The interruption that changed everything was deceptively simple. If the problem began when an Rh-negative mother's immune system first learned to recognize D, then what if you could prevent that lesson from ever taking hold? Researchers realized that if you gave an Rh-negative woman a small dose of anti-D antibodies from an outside source at exactly the right time after delivering an Rh-positive baby or after a miscarriage, abortion or certain procedures, you could coat any fetal Rh-positive red cells in her circulation before her own immune system had a chance to react and build memory. It sounds paradoxical, using antibodies to prevent antibodies. But the borrowed anti-D acts like a cleanup crew. It marks the stray fetal cells for quiet removal without triggering the mother's deep, long-term immune training. This is Rh immune globulin, often known by brand names like Rho-D immune globulin. Introduced in the 1960s, it rapidly became one of the great success stories of preventive medicine. In countries where it was widely adopted and properly timed, usually around the 28th week of pregnancy, and again within a short window after birth, the incidence of severe hemolytic disease of the newborn in incompatible pregnancies plummeted. Deaths that had once been expected, almost routine in certain families, simply stopped happening. If you look at graphs from that period, you see a steep decline, a curve bending almost straight downward. Entire specialties in pediatrics, devoted to exchange transfusions for Rh babies, became quieter. Neonatal wards changed. A particular kind of heartbreak, while not entirely gone, became dramatically rarer wherever the prophylaxis was available. It was, and is, a landmark for several reasons. First, it showed that you could prevent a disease of future pregnancies by intervening in the immune events of this one. Second, it required systems, routine blood typing, record keeping, follow-up. It pushed obstetrics toward a more proactive laboratory-linked model of care. Third, it forced medicine to see pregnancy in a new way, not just as a natural process to be supported, but as a place where microscopic incompatibilities could be anticipated and diffused. And then there is the social layer. When public health campaigns began to emphasize Rh status, when prenatal clinics started handing women cards that said Rh negative in clear print, when posters and pamphlets explained in simple language that some mothers needed an injection to protect future babies, the phrase Rh negative stepped out of the lab and into everyday consciousness. For many families, it became bound up with their most intimate stories. I lost 2 babies before they figured out I was Rh negative. They told me I needed a shot after my son was born to save my next child. My mother says the reason I'm alive is because by the time she had me, they knew about Rh. In that shift, from obscure antigen to widely known risk factor, Rh negative blood acquired an aura. It was no longer just a minus sign on a chart. It was a pivot point in reproductive fate, a tiny molecular absence around which life and death had once quietly turned. This history matters when we think about the myths that grew up later. When you know that, within living memory, being Rh negative could mean losing child after child unless medicine intervened at exactly the right moment, it is easier to see why this blood type attracted so much attention, anxiety, and eventually storytelling. Before we get to those stories, though, we need one more piece of the puzzle: how Rh negative is inherited, and why it's still here at all. To get there, we have to zoom in again, this time not on a single pregnancy, but on the quiet arithmetic happening in every conception. Start with the simplest version of the story: two Rh negative parents. On their red blood cells, the D antigen is absent. In their DNA at the RhD gene, they each carry forms that do not produce a working D protein. In genetics shorthand, you can think of it as two copies of a D allele rather than the D version. When they have a child, they can only pass on what they have. Each parent contributes one copy from their pair. If both are D, then every child gets D from one side and D from the other. There is no D in the mix to appear by surprise. The result is another Rh negative person. No magic, no purity, just recessive inheritance. Recessive here means that not having D only shows up on the surface when both gene copies say the same thing. If even one working D version is present, the red cell will carry the antigen, and the person will type as Rh positive. That's why Rh negative can hide. Imagine an Rh positive man whose genes are D divided D. One copy makes a working D antigen. The other is the silent form. His blood test only sees the antigen on the cell. It doesn't read his underlying pair. Now imagine an Rh negative woman, D. When these two conceive, every child will get one copy from each parent. From the mother, it's always D. From the father, it's a coin flip. Sometimes D, sometimes D. Put those together and you get two possibilities. D children who are Rh positive like their father on the surface, or D children who are Rh negative like their mother. From the outside, this can feel baffling. An Rh positive father, and Rh negative mother, some Rh positive babies, and then suddenly an Rh negative baby that looks like a kind of throwback. In families where no one has ever talked about blood types, it can seem like something has appeared from nowhere. It hasn't. It has simply stopped hiding. This is how Rh negative can move silently through generations. Two Rh positive parents can each carry one D without knowing it. If they are both D, then at each conception there is a small but real chance, about 1 in 4, that the child will inherit the D from both sides and be Rh negative. That child is not evidence of a mysterious bloodline suddenly inserting itself into the family. It's the visible tip of a genetic pattern that was already there, just masked by dominant D alleles in parents and grandparents. In old village gossip, this kind of thing could be explosive. Before people understood recessive traits, a baby who didn't match what others expected might raise eyebrows. Even in the era of blood typing, there were court cases and arguments built on misunderstandings of these patterns. A basic textbook cross, two carriers producing a recessive child, could be misread as proof of infidelity, or of some strange intrusion from outside the family. The reality is much calmer. Human inheritance is probabilistic, not moral. Every conception rolls the same dice, quietly ignorant of our social narratives. Once you zoom out from families to whole populations, the math takes on a different shape. Think of every person in a population as carrying 2 slots for this trait, D or D, in each place. The frequency of those slots, the proportion of all the Ds and all the Ds floating around in everyone's DNA, is what geneticists call allele frequency. If D is rare, most people will be DD. A smaller number will be DD carriers, and only a tiny fraction will end up DD and visibly Rh negative. If D is more common, the number of carriers rises, and so does the number of Rh negative individuals. Nothing about this requires selection for or against Rh negative. You can get different frequencies in different places simply by how populations grew, shrank, and split apart. Imagine a small group of people leaving a larger community, 20 or 30 individuals migrating over a mountain pass, or across a narrow sea, or into an isolated valley. Just by chance, they may carry more D alleles than the population they left behind, or fewer. This is genetic drift, random sampling like pulling a handful of coloured beads from a bowl. If your handful happens to have more blue beads than average, the little bowl you start with will have a different composition than the big one you drew from. Once that small group becomes the ancestors of thousands, then hundreds of thousands, their original skewed handful of alleles gets amplified. What started as a random quirk becomes the new normal for that region. When this happens in a particularly small founding group, we call it a founder effect. A few men and women settle a remote island, or survive a catastrophe, or establish a new community that keeps mostly to itself for generations. Their specific mix of alleles, common ones and rare ones alike, gets baked into the genetic flavour of their descendants. In this way, traits that are globally rare can be locally common without needing any special superpower. All they need is a history of small groups, isolation and growth. The same logic applies in the other direction. A rare allele can persist for a very long time even if it never confers a major advantage, simply because it is recessive. The D allele can hide in millions of D carriers, tucked away under the mask of D, showing up as Rh negative only now and then. Selection, the relentless winnowing of alleles that are very harmful, has a harder time wiping out recessive traits that mostly live in these carriers. If being DD is slightly disadvantageous in certain circumstances, but most copies of D sit harmlessly in DD people who are fine, the allele can drift along for tens of thousands of years. This is one reason rare does not automatically mean special in an adaptive sense. Human populations carry thousands of rare variants. Some cause disease in the homozygous state, like certain metabolic disorders. Some are mostly neutral, their effects so minor that natural selection barely notices. A few may be mildly beneficial in some environments, and slightly harmful in others, averaging out to "it depends." Rh negative almost certainly falls somewhere in this spectrum. It has obvious medical consequences in the specific context of incompatible pregnancy. Outside that context, its advantages or disadvantages are much less clear. And yet, our minds are not built to be comfortable with "it depends." We like stories where rare equals important. If only 15% of people in a region are Rh negative, and even fewer elsewhere, it feels natural to think they must be different in some deep way. Maybe more intuitive, maybe more resistant to certain diseases, maybe closer to some ancient, purer lineage. These intuitions borrow from patterns that are sometimes true, like sickle cell trait offering malaria resistance in certain environments, and apply them far beyond the evidence. They also lean on older ideas about blood as essence, as character, as destiny. In a world where we now know that Rh negative status can decide the fate of pregnancies unless medicine steps in, it is easy to see why people might cast it as powerful rather than random. The stakes are so high that a simple recessive allele starts to feel like a destiny marker. The actual genetic story is quieter. Rh negative persists because recessive alleles can linger, because populations are not infinitely large or perfectly mixed, and because chance events who moved where, who had children with whom, who survived an epidemic or a famine, left their imprint on the frequencies we see now. But chance is not evenly spread. The D allele is not scattered uniformly across the globe like mist. It has clusters, gradients, and hotspots. In some parts of the world, Rh negative is genuinely rare, only a few % of the population. In others, it is unexpectedly common, reaching double digits. Those patterns are the next piece of the puzzle. If Rh negative is just a recessive trait drifting along, why do some regions have so much more of it than others? What historical migrations, bottlenecks, and founder effects carved those pockets of high frequency into the map? To answer that, we have to pull the camera back again from genes to geography and trace where, on Earth, this negative sign has come to loom the largest. When you first see a global map of Rh negative frequencies, it can look almost like a weather chart. Cool colours across most of the planet, a few warmer patches, and then over parts of Europe, a brighter flare. If you averaged the whole human species into a single number, you'd say something like this. Most people on Earth are Rh positive. Rh negative is a minority status almost everywhere, but averages blur the interesting parts. In much of East Asia, for example, only a few % of people are Rh negative. In parts of Africa and South Asia, the numbers are also low, often under 10%. Move into many indigenous populations of the Americas, and Rh negative can be even rarer. Then you look at Western Europe, and the picture changes. In some populations, Ra negative frequency rises into the teens, even around 15% or a little higher. Not everywhere, not in some uniform European bloc, but in clumps. Gradients, a patchwork. Those are the clusters that get people talking. If you've ever fallen down an internet rabbit hole about Rh negative, you've probably seen maps with thick red blotches over places like the British Isles, parts of France, and especially a small region straddling the border of modern Spain and France. Those maps often come with captions that leap far beyond the data. Ancient bloodlines, pre-Diluvian survivors, non-human origin. To make sense of all this, you have to hold two ideas in your mind at once. First, yes, the geography is real. Rh negative does cluster, and some populations do carry it at higher rates than their neighbours. Second, those clusters can arise from completely ordinary processes, migration, isolation, chance, without needing any hidden civilization or secret lineage behind them. Think about how human groups actually move through space. People don't spread evenly like paint poured on glass. They travel in bands, in families, in boatloads and wagon trains and caravans. They settle valleys and coastlines, follow rivers, cling to familiar languages and marriage customs. They sometimes intermarry widely. They sometimes turn inward and keep mostly to themselves. Every time a small group peels off from a larger population and becomes the ancestor of many, the genetic dice roll again. Imagine a coastal village thousands of years ago. A storm, a dispute, or a simple desire for land pushes 20 families to sail west and settle an island. By pure chance, those families carry slightly more D alleles than the mainland average. They don't know it. They never think in terms of D and D. They just have children. On that island, over centuries, their descendants multiply. The original skew, more D in that founding group, gets magnified. A trait that was once moderately common becomes noticeably common. Maybe 1 in 5 people is now Rh negative. Now suppose contact with the mainland is intermittent. Trade exists, but marriage mostly happens within the island community and with a few neighbouring settlements that were founded the same way. You've just built a hotspot, no aliens required, no lost Atlantis, just founder effect, isolation and time. European history offers countless opportunities for this kind of process. Glacial refuges during ice ages, where small bands survived in southern pockets while northern lands froze. Post-ice age recolonization, as those survivors spread back north and west. Later, the rise and fall of farming villages, hill forts, fishing towns, mountain hamlets, plagues that emptied some regions and spared others. Wars that wiped out whole lineages in one valley and left the next valley almost untouched. Layer on top of that the habit of endogamy, marriage within a community, a faith group, a linguistic cluster, and you get little genetic echo chambers dotted all over the landscape. Each echo chamber has its own starting frequencies for alleles like D, set by whoever happened to be there when the group formed. Once the group grows and stays relatively distinct, those frequencies can drift upward or downward, sometimes dramatically. This is the math behind many of the European Reich negative clusters. Regions that today show higher percentages are often places that historically were somewhat isolated, mountainous territories, linguistic islands, communities that by geography or culture had fewer incoming marriage partners for long stretches of time. Once you start looking for the demographic story behind a hotspot, the maps feel less like evidence of a separate species and more like a record of human traffic jams and quiet side roads. They also become messier, which is closer to the truth. Those clean, high-contrast internet maps you see, the ones that paint whole countries in a single shade, are often based on sparse or outdated data. Sometimes they're extrapolated from small samples taken decades ago. Sometimes they ignore urbanization, migration, and intermarriage that have blurred older patterns. A national border on a map is not a genetic wall, yet you will often see an image that colours one nation high Rh negative and its neighbour low, as if people stopped reproducing at the customs station. Real frequency maps, built carefully from many studies, tend to show gradients rather than sharp edges. Numbers fade from one value to another across space, reflecting rivers, mountains, trade routes, and historical events, not clean, straight political lines. And even those better maps can mislead, because our eyes are drawn to the dramatic parts. A region where 15% of people are Rh negative looks visually as if it is blazing with difference compared to a region where only 5% are. On a heat map, that can be the jump from pale yellow to deep red. But pause on what those numbers actually mean. In the high region, 85 out of 100 people are still Rh positive. In the low region, 95 out of 100 are. Both places are overwhelmingly Rh positive. The local rise in Rh negative can be striking without representing some total inversion of human biology. It's a subtlety our pattern-seeking brains are bad at holding during a late-night scroll. This is where stories rush in to fill the gap. If you live in or read about a region where Rh negative is relatively common, and you've seen those bright coloured maps and heard about Rh negative pregnancies being special or dangerous, it can be tempting to read a deeper narrative into it. Maybe you think, our ancestors were different. Maybe they were here first. Maybe this cluster is the fingerprint of an ancient people that survived, while others moved on or died out. Sometimes there is a kernel of truth in that instinct. Hotspots do frequently overlap with populations that have deep continuity in a region, groups that retain their language or culture through waves of conquest and assimilation. But continuity is not the same as separateness. You can carry an older linguistic or cultural tradition and still be part of the same broad human genetic fabric as your neighbours. You can have a slightly higher local frequency of a recessive allele without that allele defining you as a distinct kind of being. The danger comes when those maps are used to tell hierarchical stories. This group as more ancient, more pure, more chosen. Or in darker corners of the internet, when they're twisted into exclusionary myths about who does or does not belong. Rh negative, by its very nature, lends itself to this. It is invisible most of the time, then suddenly powerful in the context of pregnancy. It is rare enough to feel special, clustered enough that you can draw circles around certain peoples, ancient enough that no one remembers when it first appeared. Combine that with the human tendency to romanticize isolation mountain folk as noble holdouts, islanders as guardians of secret wisdom, and the step from interesting genetic pattern to mystical bloodline is a small one. But if you strip away the romance, what you see beneath almost every Rh negative hotspot is the same trio of forces: small founding groups, relative isolation, and the slow churning of time. An allele that was slightly more common in the founders becomes more visible in their descendants. Mountains or customs or both slow the inflow of outside genes, Generations pass. Then, centuries later, someone takes blood samples, plots a map, and the old accidents of history appear as bright patches of colour. Among all those patches, one has attracted more attention than any other. It sits in a corner of southwestern Europe, where a people with a distinctive language and a reputation for endurance have, for a long time, carried a higher than average frequency of Rh negative blood. That region has become the favourite canvas for stories about ancient blood, lost civilizations and secret powers. To see how a simple cluster turned into a magnet for myth, we have to zoom in on that case on the Basque country and watch how geography, language and genetics tangle together in the modern imagination. If you trace your finger along the Bay of Biscay, where the Atlantic bites into the western edge of Europe, you'll cross a landscape of green hills, jagged coastline and mountain valleys. In that corner, between what we now call Spain and France, lives a people who have been there, in some recognisable form, for a very long time. They call themselves Euskaldunek. We call them the Basques. For most of their history, the Basques weren't famous because of blood types. They drew attention for two other reasons: where they lived and how they spoke. Geographically, the Basque country sits in a kind of natural pocket, shielded by the Pyrenees to the east, opening onto the rich fishing waters of the Atlantic to the north, bordered by different cultural zones to the south and west. Strategically placed, but also slightly off to the side of the main highways of empire. Historically, that pocket became a refuge and a crossroads at the same time. Archaeology in the wider region shows human presence going back tens of thousands of years. Caves with Paleolithic art, stone tools from hunter-gatherers who survived the last ice age in the shelter of southwestern Europe. Then later Neolithic farmers, Bronze Age traders, Iron Age warriors, Roman soldiers, medieval kingdoms. Wave after wave of people moved across Iberia and Gaul, but some local lineages, and probably some local speech, persisted. By the time written sources appear, outsiders already recognized the inhabitants of that region as distinct. The Romans and later chroniclers noted their stubbornness, their local autonomy, their different tongue. That tongue is what really sets the Basques apart in the European imagination. Basque is what linguists call a language isolate. It's not just a quirky dialect. It doesn't belong to the Indo-European family that includes Spanish, French, English, German, and most other European languages. It doesn't slot easily into any known surviving family at all. You can line up Basque words with Latin, Celtic, Germanic, Slavic, and they just stare back at you. The grammar goes its own way. The roots don't fit the common patterns. It is, as far as we can tell, the last surviving member of a once broader group of pre-Indo-European languages that were spoken in Western Europe before Indo-European tongues swept through. To modern ears, especially those used to tidy categories, that uniqueness feels almost supernatural, a lone language holding its ground for millennia surrounded by newcomers. For centuries, scholars have proposed theories about where it came from. Maybe related to ancient Iberian inscriptions, maybe a remnant of a wider Aquitanian speech zone, maybe connected, very speculatively, to some distant Caucasian languages. The honest answer, if you ask cautious linguists, is we can see some ancient regional connections, but beyond that, we don't really know. The deep origins of Basque are still hazy. Written records only go back so far. Spoken traditions leave fewer clear traces as you push deep into prehistory. That uncertainty, the open space where mystery lives, has always been a magnet for stories. When 19th and early 20th century scholars began to compare languages systematically, Basque stood out as an anomaly that survived. When 20th century genetics arrived and began sampling blood types across Europe, another anomaly emerged in the same region. In some Basque communities, the percentage of people with Rh-negative blood, specifically the DD genotype, was noticeably higher than in surrounding populations. Not subtly higher, strikingly higher, While many European populations might show Reich negative frequencies around, say, 12–15%, some studies of Basque groups reported figures creeping toward 20, even a bit more, depending on the sample and the time. If you focus on particular valleys or historically endogamous villages, those numbers could be even more pronounced. Not every Basque person, not every town, but enough to make the region stand out on the early maps. For most geneticists, this pattern wasn't mysterious in itself. It fit comfortably into the framework we've already talked about: founder effects, isolation, marriage patterns, and time. The Basque country, with its mountain valleys and strong local identity, had a long history of relative genetic continuity. People married within their communities at higher rates. They maintained distinct cultural and linguistic boundaries through Roman rule, Visigothic kingdoms, Islamic expansions, and later Spanish and French states. If the founding populations of some of those communities just happened, by chance, to carry a slightly higher proportion of the D allele, then centuries of relative endogamy could easily boost Rh negative frequencies. From a demographic point of view, it's almost textbook. Smallish population, partial isolation, deep time. You expect certain alleles to drift upward. Rh negative simply happens to be one of the more visible ones. But outside the narrow world of population genetics, the combination was irresistible. Here was a people already framed as ancient and stubbornly distinct, speaking a language nobody could neatly classify, living in a historical refuge zone associated with Ice Age survivors. And now, on top of that, they appeared on blood maps as a big red hotspot. To a narrative-hungry imagination, this didn't look like a standard founder effect. It looked like a signature. By the late 20th century, books and articles started linking these threads. Some were scholarly, cautious, trying to understand European prehistory by looking at Basque DNA as a potential window into older layers of the continent. Others were not cautious at all. In popular retellings, Basques have the highest Rh negative frequencies in the world, morphed into Basques are the original carriers of Rh negative blood, then into Basques are remnants of an ancient separate people, and finally, in some corners, into Basques, and by extension Rh negative people are descendants of a lost civilization or even a non-human lineage. The linguistic isolate became proof that they were not just culturally distinct, but biologically other. The Rh negative hotspot became evidence of a different origin story, disconnected from the rest of humanity. Notice how the leap happens. A real observation: Basque is not Indo-European. Another real observation: some Basque populations show higher Rh negative frequencies than neighbouring populations. A reasonable inference: the Basque people likely preserve some genetic and cultural continuity from pre-Indo-European inhabitants of the region. Then the jump. Therefore Basques, and especially Rh negative Basques, must be fundamentally different, either more ancient than everyone else, or descended from some exotic source. What gets lost in that jump are the more mundane, but more accurate explanations. Linguistic uniqueness does not require exotic biological ancestry. A language can survive conquests and cultural shifts if the speakers maintain strong identity, favourable geography, and enough autonomy. The Basque language likely persisted because its speakers occupied defensible terrain and navigated political pressures skillfully, not because their tongues were wired differently at birth. Similarly, a higher frequency of a recessive blood type doesn't mean the group is a different kind of being. It means that at some point in their demographic history, the relevant allele was a bit more common and then drifted upward in a relatively contained population. Genetic studies of Basques over the last decades consistently show that they are not alien to the rest of Europe. They cluster within the broader Western European genetic variation, with some distinctive features and deeper continuity, but no dramatic break. They look like what you'd expect from a population that, for thousands of years, mixed less than some neighbours and retained more of certain ancient lineages. In other words, more continuous, not more separate. But in the realm of myth-making, continuous doesn't capture the imagination the way separate does. Once a single striking statistic enters popular culture, Basques up to 30% Rh negative or whatever the quoted number is, it tends to float free of its original context. The details of sample size, specific villages, time of collection, and comparison groups fall away. That statistic gets repeated, rounded up, and embellished. It becomes the seed around which stories crystallize. Add in the Basque language's mystery, the region's Ice Age refuge history, and modern fascination with underdog cultures, and you have the perfect stage for speculation. Each piece is real enough on its own. Together they create a pattern our brains are almost helpless not to mythologize. Some versions of the story are harmlessly romantic. Basques as guardians of ancient European genes, Rh negative as a marker of deep time. Others slide into more charged territory, Basques as purer, more original, more noble, Rh negative as a sign of chosenness, outsiders as diluted or less authentic. Those ideas can feed into nationalist or exclusionary narratives that have little to do with biology and everything to do with human politics. The same pattern shows up in New Age and fringe communities, but with a cosmic twist. Basques become stand-ins for Atlantean survivors or star ancestors. Rh negative people everywhere are invited to see themselves as inheritors of that special lineage, regardless of where their actual grandparents were born. All of this grows from a simple starting point. One cluster on a map, one unusual language, one region with a reputation for persistence. The Basque case shows how easily a legitimate demographic pattern can be lifted out of its historical and genetic context and turned into a canvas for whatever story someone wants to tell. It doesn't mean there's nothing interesting going on. Basque history really does tell us about deep layers of European population structure. Their language really is a rare survivor. Their Rh-negative frequencies really do sit on the high end of the human spectrum. But interesting is not the same as supernatural. Distinct is not the same as separate. And continuity is not the same as purity. If you can keep those distinctions in mind, the Basque example becomes a kind of cautionary tale. It shows how quickly a real statistic, mixed with a bit of mystery, can sprout decades of speculation about bloodlines and origins. And it prepares us for what comes next. Because once you accept the idea that a blood-type cluster in one European region might hint at a special lineage, it's a short step to asking why that lineage exists at all. Maybe people begin to suggest Rh negative itself is the trace of some ancient selection pressure, some hidden advantage, some almost magical trait. In the next part of the story, we'll look at those evolutionary theories, the sober ones, the speculative ones, and the blank spaces where science still doesn't have definitive answers. To talk about evolutionary theories of Rh negative blood, you have to start with a simple question that sounds almost too blunt. Did this thing actually help anyone survive? Because under all the romance and mystery, evolution is not sentimental. Traits spread, or they fade, because of birth and death. Who lived long enough to have children? Who didn't? So imagine for a moment a world in which Rh really did matter in some concrete way. In that world, being Rh negative would either make you more likely to survive and reproduce in certain conditions, or less likely. Evolution doesn't care about symbolism, it cares about numbers. If Rh negative was strongly harmful, you'd expect it to be squeezed down to almost nothing over tens of thousands of years. Not gone completely recessive alleles can hide, but very rare. If it conferred a big benefit, you'd expect the opposite. It would spread, especially in populations where that benefit applied. Maybe you'd see whole regions where almost everyone was Rh negative. The way you see high rates of lactose tolerance where dairying was favoured, or malaria-resistant genes where mosquitoes were a brutal filter. What we actually see is something in between. Globally, Rh negative is a minority. In many populations, especially in parts of Africa and Asia, it's quite rare. In others, particularly in Europe and some nearby regions, it's more common but still not a majority. Even in those high-frequency pockets, most people are still Rh positive. That distribution doesn't scream superpower. It looks more like a trait that can drift up or down, sometimes nudged by local conditions, sometimes just riding along with demographic history. But there is one place where Rh status clearly does matter in life and death terms: pregnancy. This is where evolutionary thinking gets sharp and a little uncomfortable. When an Rh negative woman carries an Rh positive fetus, her immune system can, under certain circumstances, treat some of the fetus's blood cells as foreign. Not usually in the first pregnancy, but if fetal cells slip into her bloodstream and her immune system responds, she can become sensitized. In later pregnancies with Rh-positive fetuses, her antibodies can cross the placenta and attack the baby's red blood cells. Before modern medicine, that could lead to severe disease in the newborn. Brain damage, stillbirth, maternal grief repeated across generations. So one obvious question emerges: if Rh-negative mothers face that risk in a world without anti-D injections and blood screening, why didn't evolution wipe the allele out? There are a few important wrinkles here. First, the conflict only appears when an Rh-negative mother carries an Rh-positive child. If both parents are Ra negative, there's no problem. If the father is Rh positive but carries the D allele, some children are Ra positive, some are Rh negative. So the risk is real, but it's not universal. Second, the timing matters. The worst effects often hit later pregnancies after sensitization. In many historical societies, a woman's first few children might already carry her genes forward before the most severe immune reactions become common. Still, across thousands of years, even a moderate increase in infant death would put pressure on a trait. So evolutionary biologists and geneticists have asked, is there any hidden benefit that might balance this danger? One family of hypotheses centers on something called maternal-fetal conflict. Pregnancy is not a peaceful union of interests. It's a negotiation. The fetus, carrying genes from both parents, wants more resources, more nutrients, more blood flow than the mother can always safely give. The mother's body, shaped by her own survival odds, wants to support offspring, but not at the cost of severe damage or death. In that tug-of-war, immune recognition is one of the tools. Some researchers have suggested that Rh differences might slightly alter this balance. Perhaps certain Rh combinations shift how blood is exchanged, or how strongly the mother's body regulates the placenta. Maybe, in some environmental conditions, scarce food, high disease loads, certain Rh pairings made pregnancies a little more successful, or improved maternal survival, even if there were costs in other pairings. This is an elegant idea, but at the moment it's more sketch than finished portrait. The evidence is patchy, sometimes contradictory. Studies are complicated by modern healthcare, by changing diets, by regional differences in ancestry. Another set of hypotheses looks outward, not inward, toward pathogens. The Rh proteins are embedded in the membrane of red blood cells. We still don't fully understand their original biological function, but they seem to be involved in moving certain molecules, maybe gases like ammonia, across the cell membrane. When you change the shape or presence of a protein that sits on the outside of cells, you also change how pathogens can interact with those cells. Some parasites latch onto very specific structures. Some bacteria and viruses exploit particular receptors to gain entry or to hide from the immune system. So it's not crazy to ask, could Rh-negative blood, with its altered surface, have made some people slightly more resistant or slightly more vulnerable to particular infections? A few epidemiological studies have hinted at differences. Some report small associations between Rh status and risks for certain conditions. There have been suggestions, for instance, of links between Rh-negative status and susceptibility to particular parasites, or subtle differences in certain disease outcomes. But hinted at is the key phrase. These are not ironclad. Repeat everywhere results. They vary from study to study. Many have small sample sizes. Others struggle to separate Rh status from all the other factors that influence health, income, nutrition, other genes, local healthcare quality. So we're left with a cloud of possibilities. Maybe Rh-negative offered slight protection in some disease environments. Maybe it carried small disadvantages in others. Maybe in some places those tiny shifts mattered enough over long periods to nudge frequencies up or down a little. This is where the idea of trade-offs becomes important. In evolution, a trait does not need to be universally good or bad. It only needs to tilt the odds in a particular context. Sickle cell trait is the classic example. One copy of the gene can confer protection against severe malaria. 2 copies cause sickle cell disease, which can be devastating. The same variant is both life-saving and life-threatening, depending on how many copies you inherit and where you live. Rh-negative doesn't behave in exactly the same way, but the logic is similar. A trait can persist even if it causes serious problems in some circumstances, as long as those problems are limited, delayed, or offset by benefits elsewhere. And traits can persist even with no real benefit at all, simply because of drift, especially when they're recessive and mostly hidden in carriers. In that sense, Rh-negative might be less like a sharp tool tuned by selection and more like a slightly odd stone that ended up in the mosaic of human variation because nothing ever pushed it strongly out. When scientists try to decide which story is closer to the truth, they lean on different kinds of evidence, each with its own limits. They look at population frequencies, where the allele is common, where it's rare, how that maps onto historical migrations and known disease environments. They run statistical tests for signals of selection in the genome, looking for patterns that suggest an allele has been favoured or disfavoured. They analyse medical records, searching for correlations between Rh status and disease outcomes, knowing that correlation is not causation and that many hidden variables lurk in the background. They build models of how incompatibility would affect birth and death rates in pre-modern societies, then see if those models match real-world frequencies, and they argue with each other, politely in journals, sometimes less politely at conferences, because the data are noisy, and history is long, and the human body is complicated. What comes out of that process is not a cinematic revelation. Rh-negative was the warrior's blood, or the healer's blood, or the star-born blood, but something more modest. We know Rh-negative can create dangerous conflicts in pregnancy when not managed medically. We know its frequencies vary around the world in ways that partly follow ancestry and drift. We have some speculative partial hints that it may have interacted with disease environments or with subtle aspects of maternal-fetal dynamics. We do not have a clean, proven story where Rh-negative clearly made its carriers stronger, smarter, more psychic, more empathic, or more anything in a simple, one-directional way. That gap between what we'd like to know and what the evidence can support is where culture moves in. When people talk about Rh-negative as a superpower, they are, often without realizing it, filling in that gap with meaning. They see a minority trait, a history of risk in pregnancy, a scattering of mysterious maps and Basque statistics, and they read into it a sense of specialness. Sometimes that specialness is a way to reclaim something frightening. If a blood type has been associated with danger to babies, turning it into a mark of higher intuition or ancient lineage can be comforting. It transforms vulnerability into destiny. Sometimes it's just another version of an old human habit, taking rare biological differences and wrapping them in stories of chosenness, purity, or secret power. From the perspective of evolutionary biology, though, Rh-negative is not a verdict. It's a variation, one thread in a much larger fabric woven by selection, chance, migration, and time. The real mystery is not why some people spin superpower narratives around it. That part is very human. The mystery is what exactly Rach negative was doing as human populations moved through ice ages, epidemics, famines, and centuries of childbirth without modern medicine. On that question, science still has more questions than answers. And into that quiet, into that uncertainty, other voices have stepped. Voices that are less interested in maternal-fetal conflict curves and more interested in lost continents, fallen angels, and visitors from the stars. In the next part, we'll watch how those evolutionary questions get taken up, stretched, and transformed into full-blown origin myths for Rh-negative blood. By the time the 20th century really got going, all of these deep evolutionary questions about Rach were buried in silence. Nobody was arguing about maternal-fetal conflict curves. Nobody was mapping allele frequencies across continents. Most people didn't even know blood came in types. If you were a farmer in 1900, or a factory worker or a schoolteacher, the idea that your blood might be different from your neighbour's in some invisible, medically important way, that just wasn't part of everyday thought. Blood was blood. The universe had other plans, because when humans industrialized war, they also accidentally industrialized blood. The First World War was one long, brutal experiment in what happens when millions of bodies meet new forms of destruction. Machine guns, artillery shrapnel, high explosive shells. Soldiers bled out in trenches, in field hospitals, in makeshift aid stations under canvas and mud. Surgeons already knew that transfusing blood could sometimes save lives. They also knew, from bitter 19th century experience, that it could just as easily kill. Without typing, transfusions were a coin flip with a loaded coin. Pour the wrong blood into a wounded soldier and their immune system might attack it violently. Red cells clump, vessels clog, kidneys fail, death comes fast. So while scientists in laboratories were mapping out the basic ABO system in the early 1900s, the trenches forced medicine to turn that abstract knowledge into procedure, match the type, or don't transfuse at all. Field units started to test blood. Crude at first, then more systematic. Even then, the focus was on the big visible reactions, the obvious incompatibilities, the dramatic collapses. The Rh factor, the quieter culprit that could create delayed, sometimes deadly reactions, was still in the shadows. It had been noticed in experiments, argued about in papers, but it wasn't yet burned into protocol. That would change with the next, even larger catastrophe. By the time the Second World War began, blood transfusion had moved from a dangerous curiosity to a recognized, life-saving tool. The problem was scale. This wasn't about saving a handful of patients in a city hospital anymore. This was about entire armies, cities under bombing raids, civilian casualties by the tens of thousands. You couldn't just tap a donor lying next to the wounded man on a stretcher and hope for the best. You needed blood stored, labelled, moved, ready, so governments did something new in human history. They built blood banks. Refrigerators, anticoagulants, standardized bottles, trucks and trains carrying units from collection centres to battlefronts. Campaigns that urged civilians to do their part by rolling up their sleeves. With those banks came another innovation, the idea that your blood type was a piece of personal data worth knowing. Typing stopped being just a laboratory curiosity. It became a line on a card. In Britain, in the United States, in parts of Europe and the Soviet Union, people signed up for donor registries. Nurses pricked fingers. Technicians spun tiny drops in test tubes with reagents. Results were written on paper files, metal tags, wallet cards. Name, address, a bow type. At first, Rh wasn't always there. The factor had only been firmly described near the end of the 1930s, and wartime systems are slow to change. But clinical experience has a way of sharpening attention. Doctors began to see it. Some compatible transfusions matched by ABO still went wrong. Hemolysis, fevers, subtle patterns that hinted at another layer of compatibility. The Rh factor moved from the journal page into the hospital chart. By the late stages of the war, and especially in the years after, blood typing meant two things side by side: your ABO group and your Rh status. A, B, AB or O, positive or negative. Those little plus and minus signs, those capital letters, would eventually crawl off the lab sheet and into the public imagination. But before they could do that, they had to become routine. So after the war ended, the machinery built for conflict found new targets: peacetime medicine, public health, maternity care. Hospitals kept their blood banks. Civil defense plans in many countries included stockpiles of typed blood. Donor drives continued, sometimes framed as preparation for the next possible war, sometimes as a civic duty in calmer times. Blood type knowledge spread outward. It showed up in basic health education, in school biology classes, in pamphlets handed out at clinics, in the fine print of insurance forms. People started to say sentences that would have been strange a generation earlier. I'm O positive. They told me I'm AB negative. For most, it was a little bit of trivia, a fact you might mention once and then forget. For some, it became something else. Part of what made that transformation possible was what was happening at the other end of life, in maternity wards. Because while war medicine had pushed forward transfusion science, peacetime medicine had been quietly wrestling with a recurring horror: hemolytic disease of the newborn, often caused by Rh incompatibility. These weren't wounded soldiers, these were babies. Some women, especially those with Rh-negative blood, would carry healthy-looking pregnancies, then deliver infants who turned jaundiced, anemic, or died soon after birth. Others had multiple stillbirths with no clear explanation. Doctors noticed patterns. They compared charts. They saw that mothers with certain blood types seemed at higher risk when the father was Rh positive. The invisible molecular conflict that had been playing out in wombs for as long as Rh variation existed was finally being translated into clinical language. In the mid-20th century, as the Rh factor became part of routine prenatal care, this knowledge was weaponized in a different way: prevention. Testing pregnant women for Rha status, monitoring antibody levels, matching transfused blood with more care. Eventually, in the 1960s, developing the anti-D injection that could prevent sensitization in Rh-negative mothers carrying Rh-positive babies. For the families involved, this was nothing short of miraculous. A sentence of recurring grief was commuted. Later, children who might have died were born healthy. And for those women, Rh status was no longer an abstract lab result. It was an intimate, high-stakes fact about their bodies, a reason for extra appointments, a note in their medical record, a conversation with their doctor about what might happen to their child. So, by the middle of the 20th century, Rh had become a practical concern in two of the most emotionally loaded places in modern life: on the edge of death in the operating room, and at the threshold of birth in the maternity ward. That is precisely where medical facts tend to leap into culture. Media followed. Newspapers ran human interest stories about miracle babies saved by new blood tests. Magazines explained blood types in friendly, simplified diagrams. Radio programs and later television segments talked about the importance of knowing your type in emergencies. Public health campaigns encouraged people to carry donor cards, to write their blood type on their ID, even in some countries, to have it stamped on military dog tags. The message was straightforward. This information could save your life, or your child's life. Behind that simple slogan, a more subtle shift was happening. Blood type, and especially being in a rare category, was starting to feel like a label, a category, a small medically defined box you could step into. Most people learned at some point that O negative was the universal donor, able to give to almost anyone. They heard that AB positive was the universal recipient. They might hear that AB negative or B negative were very rare. Hospital posters colour-coded these facts. Charts in clinics grouped people by percentages. X% of the population is this type, only Y% is that type. If you were one of the few who fell into a small slice, O negative, AB negative, or, in later conversations, RH negative more broadly, you were suddenly aware that you were outnumbered. Not in a cosmic evolutionary sense. Just in a simple arithmetic way, you were a minority category in a system that now mattered for health. Modern societies are very good at turning minority categories into identities. Think of how left-handedness, once punished or ignored, became a small badge of difference. Think of how being colourblind, lactose intolerant, or allergic to peanuts can become part of how people describe themselves. Medicine, by naming and tracking these differences, unintentionally hands people a vocabulary of uniqueness. Ride negative status slotted neatly into that pattern. You could be told in a calm, clinical voice that only a small percentage of people are Rh negative. You might be given extra monitoring in pregnancy, or asked to donate blood because your type was in short supply. Those experiences carried a quiet implication. You are not like most people, at least in this one invisible way. In a world increasingly hungry for micro-identities, where personality quizzes, astrological signs, and psychological labels all compete to explain who we are, that little biomedical distinction had all the right ingredients to become meaningful far beyond the hospital. It was numerically rare, medically important, and easy to state in a sentence. By the late 20th century, as mass media turned into global media, those sentences could travel. An article about Incompatibility in a parenting magazine in one country could be read by someone halfway around the world. Online forums began to collect stories, women sharing their experiences of being Rh negative, donors talking about being called in for special drives, patients recounting odd comments from doctors about their rare blood, pieces of correct information mingled with half-remembered lecture notes and dramatic personal anecdotes. This blood can save everyone. This blood is endangered. This blood is different. Once you have a story seed like that, difference, rarity, high stakes, and you plant it in the cultural soil of the late 20th and early 21st centuries, where identity is constantly being invented and reinvented, something predictable happens. The language of medicine starts to blur into the language of meaning. Rh negative stops being just an antigen status. It starts becoming a kind of shorthand people use in conversation about who they are, where they come from, what makes them special. And wherever you have a label that feels special, that feels rare, that feels a bit mysterious, even to experts, you have the perfect ground for legends to grow. In the next part, we'll watch how that quiet clinical category, born in war hospitals and maternity wards, was reimagined as a mark of destiny, a secret lineage, even a leftover trace of otherworldly ancestors. When people first started saying, "I'm Rh negative," they weren't making a cosmic claim. They were usually repeating something a nurse or doctor had mentioned in passing. But brains are pattern-making machines, and they hate loose ends. If you're told your blood is rare, that doesn't just land as a lab result. It lands as an open question. Rare how? Rare why? Rare for what? Rarity is fuel for imagination. We see it with eye colours, with double-jointed thumbs, with unusual allergies. Any trait that shows up in a small slice of the population invites stories. Maybe it's special, maybe it comes with hidden advantages. Maybe it explains why you feel different from the people around you. Rh negative had all the right ingredients for an extraordinary narrative. It was statistically uncommon in most populations. It was tied to very dramatic real-world events, life and death transfusions, pregnancies on the edge. It showed clear patterns on maps, clustering more in some regions than others. And crucially, scientists themselves could not yet fully explain its long-term evolutionary story. Rarity, high stakes, geographic mystery, scientific uncertainty. That combination is like a magnet for meaning-making. So, as more people learned their status, a quiet, familiar question started to surface in living rooms, online forums, late-night conversations. If my blood is different, am I different? Not in some vague philosophical sense, in concrete, everyday traits. This is where the secret superpower narrative began to take shape. It didn't start with grand claims about ancient aliens or forgotten civilizations. It started smaller, almost shyly, with lists. People began to compare notes. I'm writing a RET negative and I've always had lower blood pressure. I'm writing a RET negative and I can't stand fluorescent lights. My mother and I are both Rh negative and we have the same weird temperature sensitivity. Someone else would read that and think, wait, that sounds like me. A pattern, or the illusion of one, would form. Over time, certain themes bubbled up again and again in these conversations. If you browse through Rh negative forums, blogs and social media posts from the early internet era, you see remarkably similar lists repeated across countries and languages. Heightened intuition, stronger empathy, unusual dreams, sensitivity to medications, feeling out of place, low resting body temperature, higher incidence of green or blue eyes, better night vision, difficulty tolerating crowded environments. Almost none of these traits have been robustly tied to Rh status in controlled studies. Many of them are common human experiences, scattered widely across all blood types. But on the level of personal narrative, they felt like puzzle pieces snapping into place. The logic, more emotional than scientific, went something like this. I have always felt a bit different. I've just been told that, biologically, I am part of a rare group. Here is a list of negative traits, some of which sound exactly like me. Therefore, the reason I feel different is my blood type. This is confirmation bias at work in its most human form. You start from a belief, or even just a hope that your difference has an explanation, and then you selectively collect evidence that supports it. You remember the hits, forget the misses, and unconsciously adjust your memory to fit the emerging story. If the list says vivid, unusual dreams, you remember the dream you had at 13 that felt like a prophecy. You don't think about the thousands of completely ordinary dreams in between. If the list says sensitivity to heat, you recall the time you got dizzy in a summer crowd. You don't weigh it against all the Rh-positive friends who also wilt on hot days. Stories spread faster than statistics, so as more of these trait lists circulated, they began to harden into a kind of folk taxonomy. Rye, negative people are like this. Rh-positive people are like that. The term superpower began to appear in headlines and posts, not always literally, but close enough. Do Ra negatives have psychic abilities? The rare blood type linked to higher intuition? Ra negative, the empaths among us? In a culture saturated with superhero movies, personality quizzes, and self-help language about finding your unique gifts, this framing made intuitive sense. Your blood wasn't just a risk factor in pregnancy or a precious resource for transfusion. It was a clue to hidden abilities. Once that door opens, almost anything can walk through it. Rh negative became linked, in different corners of the internet, to heightened pain tolerance and lower pain tolerance, depending on which thread you read, greater resistance to certain illnesses and greater vulnerability to others, faster reaction times, slower aging, better spatial awareness, more left brain or more right brain, depending on which era's pop neuroscience was fashionable. The specifics shifted. The underlying structure stayed the same. Take a rare biological marker, add a set of desirable, somewhat vague traits, invite people to see themselves reflected in those traits. Then encourage them to attribute the connection to the rare marker. The result is sticky. It feels empowering. It provides a ready-made script for who you are, and the internet amplified it. Before the web, if you were Rh negative, you might meet a handful of people with the same status in your entire life. You'd compare experiences over coffee or in a waiting room, and the conversation would end there. Once forums, mailing lists, and later social media came along, thousands of Rh negative individuals could cluster in one place, virtually. What had been a statistical minority scattered across cities and countries suddenly felt like a community. People created usernames with their blood type embedded. They formed groups specifically for Rh-negative sensitives or Rh-negative empaths. They swapped stories about feeling different, about odd medical reactions, about strange coincidences. Each anecdote was like a vote in favour of the narrative. 5 people say they hate bright lights. Interesting. 50 say it. Compelling. 500 say it in a Facebook group dedicated to the idea that Rh-negative people are extra sensitive. By that point, it feels like overwhelming proof. You don't see the silent majority of Rh-negative individuals living completely ordinary lives, never joining such groups, never thinking twice about their status. They're invisible to the story. What you see overwhelmingly are the people for whom the narrative resonates enough to seek each other out. This is selection bias layered on top of confirmation bias, and it's not a moral failing. It is how human communities often form: shared identity, shared experiences, shared language. In that sense, negative superpower culture is not that different from astrology, Myers-Briggs types, or even regional stereotypes. You start with something arbitrarily assigned or loosely correlated. You build a rich vocabulary of traits around it. You use that vocabulary to make sense of your relationships, your choices, your quirks. There is comfort in this, a feeling of being seen, a sense that your strangeness is not random. It belongs somewhere. For many Rh-negative people, seeing their experiences mirrored in others was genuinely soothing. It said, "You're not alone. There are others like you." There's nothing wrong with that comfort. The difficulty comes when comfort is mistaken for evidence. Feeling unique does not, by itself, demonstrate that a particular blood antigen is the cause. Seeing your personality described in a list does not, by itself, prove that the list is biologically grounded. Enjoying the poetry of a story does not make it a scientific finding. But humans are not built to live on pure data. We like our knowledge salted with mystery. Rh-negative status sat at a crossroads where those appetites could meet, a real, measurable biological difference, tied to dramatic medical history, wrapped in genuine scientific questions about why it persists. If you tilt that picture just slightly, you can see why the language of superpowers feels natural. A tiny molecule on your red cells, invisible to the naked eye, can decide whether your body rejects or accepts someone else's blood. It can shape the way your immune system reacts during pregnancy. It can determine whether a simple transfusion is life-saving or dangerous. That already sounds like the stuff of comic books. To move from that to it also gives me heightened intuition is, from a narrative standpoint, a smaller leap than you might think. Especially when, in the background, culture is already filled with stories about chosen bloodlines, hidden abilities passed through families, marks of destiny carried in the body. You don't have to invent that pattern. It's already there in myths, in fantasy novels, in superhero origin stories. Rh, negative, just slipped into an ancient template. The few who are different, bearing a sign in their blood. Some people stop there, a gentle, almost playful sense that their type might make them a bit special, mixed with a basic understanding of the medical facts. Others go further. They follow the trail of difference and rarity back through time, asking not just, what does this mean for me, but what does this say about where I come from? If this blood is uncommon, did it arise in some unusual group? If it clusters in certain regions, does that mark an ancient lineage? If science doesn't have clear answers yet, is there room for other explanations? This is where the superpower narrative begins to merge with something larger and stranger, stories of lost civilizations, non-human ancestors, star-born souls. The quiet identity label from a maternity ward starts to be recast as a genetic fingerprint of ancient beings. The clinical Rye negative becomes, in some circles, a hint of Atlantean heritage, Nephilim descent, or extraterrestrial origin. In the next section, we'll follow that transformation into its most elaborate form, where blood type becomes not just a medical fact or a personality mirror, but a passport to entire alternate histories of the human species. Once you start asking, where did this come from? The imagination doesn't stay small for long. It doesn't just wander back a few generations to grandparents and great-grandparents. It reaches for deep time, for origins before origins. For stories that begin with long ago before recorded history. For some people, Rh negative becomes the key that unlocks that door. One path leads to Atlantis. In these versions, Rh negative blood isn't just a quirk of human genetics. It's a leftover signature from a hyper-advanced civilization that supposedly flourished tens of thousands of years ago and then vanished beneath the sea. The idea usually goes like this. Atlantis, imagined first by Plato as a cautionary philosophical tale, is reinterpreted as a real super-civilization. Its people had higher technology, deeper spiritual knowledge, maybe even psychic abilities. When disaster struck, flood, comet, pole shift, take your pick, a small group survived and spread across the world. Their descendants, the story says, carried traces of Atlantean power in their bodies. Not in obvious ways, not glowing eyes or telekinesis. In something subtle and invisible, in the blood. Rh negative becomes, in this telling, the last biological whisper of Atlantis. Especially when people point to regions like the Basque country, or the fringes of the Atlantic and say, look, the map lines up. High Rh negative rates near the ocean, once crossed by Atlantean refugees. There is no archaeological or genetic evidence for a literal Atlantis civilization dispersing its bloodline across Europe. The Atlantean narrative is built from a philosophical allegory, 19th century occultism, and a modern taste for lost worlds. But as a story template, it is powerful. Another path runs through religious texts. In some circles, Ra negative is linked to the Nephilim, the mysterious giants or mighty men mentioned briefly in the Hebrew Bible. According to certain later traditions and interpretations, the Nephilim were the offspring of sons of God and daughters of men. Over time, this ambiguous passage has been expanded in apocryphal writings and modern speculation into a tale of hybrid beings, part divine, part human, or, in more contemporary language, part extraterrestrial, part terrestrial. Here, Rh negative blood is cast as the lingering genetic mark of that mixing. The logic often sounds something like this. If most humans are Rh positive and Rh negative can cause complications between mother and fetus, then perhaps Rh negative is incompatible with normal humanity because it comes from somewhere else. That somewhere else is then mapped onto biblical language. Fallen angels, heavenly visitors, Nephilim lineages. The rhesus antigen becomes, in effect, a theological border marker. Those without it are imagined as closer to the other side. Again, there is no credible genetic evidence that any human blood type descends from non-human beings, angelic or otherwise. Ra negative and Ra positive are normal variations within the human species, explainable through mutation and inheritance. But the Nephilim story offers something science does not, a sense of cosmic drama. a role in a very old narrative about mixing, transgression, and chosen lineages. A third path looks upward, past mythic lands and ancient scriptures into space. In these starseed or extraterrestrial origin stories, Reich negative blood is interpreted as proof that some humans aren't entirely from Earth. They are portrayed as souls or lineages seeded here by advanced alien civilizations. Pleiadians, Arcturians, Syrians. The names change depending on the source. The pitch is often soft and spiritual. If you have Reich negative blood, you may be a starseed. You might feel alienated because your soul remembers a different home. Your heightened empathy, your sensitivity, your strange dreams, these aren't random. They are signs of your off-world heritage. Here, blood type, personality traits, and metaphysical identity get braided tightly together. The language borrows from real genetics, words like DNA, hybrid, genetic upgrade, but uses them metaphorically or in ways that don't match how those terms function in biology. Charts of star systems and lists of Rh-negative traits sit side by side, inviting the listener to connect dots that feel meaningful even when the lines are not backed by data. If we pull back for a moment, we can see the pattern beneath all three of these stories. Atlantis, Nephilim, starseeds, different settings, same basic structure. A rare blood type is framed as a remnant of an earlier, superior, or at least fundamentally different population. That population is cast as wiser, more powerful, more spiritually advanced, or more technologically gifted than ordinary humans. Modern carriers become the dispersed heirs of that lost group, moving through a world that has forgotten its true history. This structure is not unique to Rh-negative narratives. It is as old as storytelling about lineage itself. You can see earlier versions in royal genealogies that claim descent from gods. In medieval Europe, kings traced their lines back to Trojan heroes or biblical figures, not because the paperwork checked out, but because it granted an aura of destiny. In Japan, emperors were said to descend from the sun goddess. In parts of Africa, rulers traced ancestry to spirits, founders, or legendary culture heroes. Blood, in these narratives, isn't a fluid carrying cells. It is a thread tying present to mythic past. For aristocracies, this mythology justified power. For marginalized or scattered groups, lineage myths could do something different. Bind a people together, offer dignity, explain survival. Stories of noble blood, chosen blood, or cursed blood have been attached to families, castes, tribes, and nations for millennia. The modern Rh-negative myths are, in that sense, familiar. They are just playing out on a new stage, with new vocabulary. What changes in the last century is the language available. We no longer speak primarily of humours and essences. We speak of DNA, chromosomes, alleles, haplogroups. So when people reach for a mythic explanation of difference, they now reach through genetics. Atlantis doesn't just pass down spirit, it passes down altered genes. Nephilim don't just bequeath reputation, they leave a measurable blood marker. Starseeds don't only carry memories, they carry encoded light in their DNA. The scientific terms are real. The way they are woven into these stories often is not. But that weaving is understandable. There is a deep human impulse to reconcile ancient frameworks of meaning with modern tools of explanation. If your sacred text says there were beings who were not like us, and your doctor tells you your blood is unlike most people's, it is not hard, emotionally, to connect those dots. If you grew up with tales of lost continents or sky gods descending, and later learn about rare alleles and population bottlenecks, it can feel natural to blend the two into a single spanning narrative. These stories do important emotional work. They give people who have felt strange or isolated a sense of belonging to something vast and purposeful. If you've never quite fit in, my ancestors were from Atlantis, or I'm part Nephilim, or I'm a starseed, can transform loneliness into destiny. The feeling of being out of place shifts slightly toward the feeling of being out of time or out of planet, and therefore special. They also flatten chaos into pattern. History, as we've seen, is full of climatic accidents, migrations forced by drought, adaptations hammered out over hundreds of generations. It is messy, contingent, often brutal. Myth cleans that up. Instead of a long chain of small changes, you get a single dramatic event, a flood, a fall, an arrival from the stars. Instead of gradual selection shaping a trait like Rh negative status, you get a deliberate act, a divine mixing, an alien experiment, a civilization choosing to encode its legacy. For a mind trying to make sense of rarity and uncertainty that can be soothing. These narratives are also sticky because they interface easily with the internet. Forums that began with gentle trait lists can slide, over time, into threads about Atlantean light codes in Rh negative DNA. Videos about Nephilim genealogy can be recommended alongside basic explainers about blood types, blurring the line between education and speculation. Once a person has had the experience of saying, "This explains me," it is very hard to let go of that explanation, even when the supporting evidence is thin. That doesn't mean people who hold these beliefs are foolish. It means they are human, using the tools available, ancient texts, modern science terms, personal feeling, to construct a coherent story of self and origin. The risk comes when metaphor is mistaken for mechanism. When Atlantean bloodline stops being a poetic way of saying, "I feel different," and starts being treated as a literal, testable heritage that overrides what genetics actually shows. When starseed DNA is used not as a spiritual metaphor but as a reason to distrust medicine or reject basic biology. So how do we live with these stories without being captured by them? How do we respect the emotional needs they speak to, while still asking calmly what the evidence can and cannot support? That is where we turn next. In the following section, we'll set aside the allure of lost continents and celestial lineages and build a simple toolkit for evaluating origin claims, about Rh negative blood, and about human ancestry more broadly. Not to drain the mystery out of existence, but to put some gentle boundaries around it, so that wonder and evidence can sit together without one erasing the other. Let's start with a very simple idea that quietly controls almost everything we can say about the past. Just because two things appear together doesn't mean one caused the other. Correlation is, these things show up at the same time or in the same place. Causation is, this thing made that thing happen. With Rh negative blood, the internet is full of correlations that are treated as causes. You might see a map of Rh negative frequency in Europe and a map of megalithic sites and notice some overlap. From there, the story writes itself. People with Rh negative blood must have built the megaliths. Their special brains gave them the ability. Their ancient civilization spread stones and genes together. But that is a leap. Rh negative is more common in some regions of Europe. Megaliths are more common in some regions of Europe. Both patterns were shaped by long, complicated histories of migration, geography, trade, and culture. They can overlap without one being the secret engine of the other. You could, with enough determination, find a similar overlap between Rh negative percentages and cheese production, or wind patterns, or the popularity of a certain saint. The map would look convincing. The story would sound smooth. The cause would still be imaginary. Even when a correlation is real and repeated, it doesn't tell you which way the arrow points. Imagine a small study that finds more people with Rh negative blood report feeling like outsiders or highly empathetic. You could interpret that as Rh negative blood causes these feelings, but it could just as easily be people who already feel different are drawn to online communities about rare traits, which talk a lot about Rh negative blood and its supposed emotional effects. They then reinterpret their feelings through that lens and report them more readily. In other words, the story might be shaping the correlation, not the blood. A calm toolkit begins by slowing that leap. When you hear, negative people are more X or Rh negative people tend to Y, you can quietly ask, is this just two things appearing together? Or is there a plausible mechanism, grounded in biology or history, that would link them? And if there is a mechanism, is there independent evidence that it's real? That word independent is important, because it brings us to what genetic studies can actually do. Modern genetics is very good at certain tasks and very bad at others. It is good at measuring ancestry signals in large groups of people. When scientists look at thousands of genomes, they can see patterns which populations share more recent common ancestors, where there were bottlenecks, where there were migrations, who mixed with whom, and roughly when. They can sometimes trace particular traits to particular events. A gene variant that helps with high altitude living in Tibet. A mutation that spread quickly because it protected people against a disease. These are signals, consistent patterns in DNA that line up with archaeological evidence, climate records, and historical sources. What genetics cannot do is read cultural stories directly out of your chromosomes. Your DNA doesn't tell you if your ancestors considered themselves Atlanteans or Nephilim, or simply members of a valley clan that liked red ochre and shell beads. It doesn't carry labels like Starseed or priestly caste or shipbuilders of the Western Sea. Those are narrative overlays. A scientist can say this cluster of people shares ancestry with this older population that lived here 8,000 years ago. They cannot say, using genetics alone, and therefore they are the rightful heirs of Atlantis. They can say Rh negative appears to be more common in some populations with deep roots in Western Europe. They cannot say, and therefore Rh negative is proof of a non-human bloodline. Consumer DNA tests sit right at this boundary between what genetics can do and what stories want from it. You spit in a tube, send it off, and a few weeks later you get percentages. 14% this, 27% that, a colour-coded map of the world. The numbers look exact. They feel almost like blood work, measured, clinical, definitive. But those percentages are not measuring Rh status. They are not saying, this part of you is Rh negative Basque, and this part is Rh positive Atlantean. They are mostly using your DNA to guess which modern or ancient reference populations you most resemble. Different companies use different reference panels and different algorithms, which is why people often get slightly different breakdowns from different services or from the same company over time as the database grows. The test might say 20% Iberian on one website, 8% French and 12% Southwest European on another, and some unknown European 10 years later when the categories get updated. None of that changes your Rih status, which is usually determined by a small number of genes that these ancestry reports don't even highlight. Rh negative is not a secret percentage hiding in your results. It's a straightforward blood type determined by whether you carry functioning copies of a particular gene for the D antigen on red blood cells. If a website or video tells you to match your Rh type with your ancestry percentages to reveal hidden lineages, it's blending two different things, a medical classification and a very approximate genealogical tool. That doesn't mean the whole enterprise is worthless. It means we have to be clear about what the numbers can and cannot say. This is where the phrase extraordinary claims require extraordinary evidence earns its place in the toolkit. An extraordinary claim is one that would, if true, rewrite a lot of what we understand about biology or history. Rh negative is a normal human variant that arose by mutation and spread through certain populations is not extraordinary. It fits cleanly into everything else we know about how traits appear and persist. Rh negative is proof of a non-human lineage interbreeding with Homo sapiens, or Ra negative comes from a lost super civilization that left no other trace in the archaeological or genetic record, is extraordinary. To support that, you would need strong converging lines of evidence, not just one intriguing study with ambiguous wording, not just a chart on a website, but multiple independent teams finding the same signal in DNA, archaeological finds that line up in time and space, historical or geological records that point to the same event. You would expect the claim to withstand attempts to poke holes in it. Other researchers would criticize methods, try to replicate results, propose alternative explanations. Over time, if the extraordinary claim were true, it would emerge stronger from that criticism. That is how things like the out-of-Africa model of human origins, or the discovery of Neanderthal modern interbreeding, moved from controversial suggestions to broadly accepted parts of our story. They accumulated evidence from different angles, until the weight was hard to ignore. Most Reich negative origin myths have not gone through that process. They live in a different ecosystem where evidence is often assembled in reverse. Instead of starting with data and asking, "What does this suggest?" they start with a conclusion and go looking for anything that seems to match. You can see this in the way misinformation spreads. A chart appears online showing Rh negative rates and some other variable. There is no citation, no information about how the data were collected, no explanation of scale or uncertainty. The chart gets shared, copied, recolored, stripped of whatever context it might once have had. Within a few years it has become common knowledge in certain circles, even though nobody using it could say where it came from. Or a study is published in a scientific journal that mentions a small association between Rh type and some trait, often with lots of caveats and low confidence. Someone screenshots one line, turns it into a meme. Science proves Rh negative people have higher whatever. The uncertainties, the alternative explanations, the might and may, and within this small sample, all of that falls away. This isn't unique to Rh negative. It happens with diet studies, with brain scans, with anything that touches on identity. Our toolkit response can be very simple. When you see a dramatic claim about Rh negative blood, ask, where is this from? Is there a source? Does the source itself contain data, or is it just quoting someone else? If there's a study, can you see the whole thing, not just the highlighted sentence? Do other studies agree? Or is this one result standing by itself, being asked to carry more weight than it was built for? And then there is the human side, because often these claims are not just data points. They are someone's story of self, telling a person, "That's nonsense, you're wrong," rarely changes a belief that is doing deep emotional work. If someone dear to you says, "I've always felt different," and learning about Rh negative and Atlantis finally made sense of it, a gentler path is possible. You can start by honouring the feeling, even if you don't share the explanation. I hear that you've felt out of place for a long time. That sounds lonely. I'm glad you found something that made you feel connected. You can distinguish between the comfort a story brings and the factual claims it makes. I can see why that narrative resonates. I'm also curious what we actually know from genetics and archaeology. Would you be open to looking at that together, just to see where the lines match and where they don't? You can share your own fascination without turning it into a contest. I love that there are mysteries in our ancestry. For me, the real story, bottlenecks, migrations, interbreeding with Neanderthals, already feels pretty magical, even without Atlantis. Can I tell you about some of that evidence? You don't have to win the argument. You don't have to strip the wonder out of their world. The aim of this toolkit is not to lock the imagination in a box. It is to give it a stable floor to stand on, to say, here is what maps and genes and bones can tell us, and here is where they fall silent. In the quiet between those two, myth and meaning can still live. But when we know the difference, we are less likely to confuse a story that helps us sleep at night with a history that has to carry the weight of actual blood and bone. In the next section, we'll bring this toolkit back to the present. We'll look at what Rh negative means today in hospitals and families, in online communities and personal identity, and ask calmly what kind of lasting power it really has in our modern world. In the present day, Rh negative blood lives a double life. On one side, it is quietly, almost boringly, medical. On the other side, it is charged with meaning, community, and sometimes a sense of destiny. Let's begin with the quiet side, the one that never makes headlines because it works so well you rarely have to think about it. In the early 20th century, people could die from a transfusion simply because the donor and recipient were mismatched in ways no one yet understood. Today, before most surgeries, before many hospital admissions, your blood is typed A, B, AB or O, and then Rh positive or Rh negative. If you are Rh negative, that little minus sign doesn't make your blood magical. It makes it incompatible with Rh positive blood in certain situations. For transfusions this matters. An Rh negative person, especially someone who has already been exposed to Rh positive cells in the past, can develop an immune reaction if given Rh positive blood. The body treats the D antigen on those foreign cells as an invader and builds antibodies against it. Those antibodies can then attack future transfused cells, sometimes with serious consequences. So hospitals track Rh type the way pilots track fuel. Carefully, routinely, without romance. Blood banks know that O negative, which lacks A, B and Rh antigens, is the universal donor in emergencies. It can usually be given to almost anyone in a crisis, before there's time to test the patient. That makes O negative blood especially valuable. But here is the important distinction. Valuable is not the same thing as superior. O negative donors are prized because their cells are less likely to trigger a reaction, not because the blood itself is stronger, purer, or more advanced. Rh negative is also central in pregnancy care. If an Rh negative person is pregnant with a fetus who is Rh positive, there is a risk that fetal blood cells can cross into the parent's circulation. The parent's immune system might then recognize the D antigen as foreign and start making anti-D antibodies. In a first pregnancy, this is often not a major problem, because significant antibody production usually takes time. But in later pregnancies with an Rh positive fetus, those antibodies can cross the placenta and start destroying fetal red blood cells. This is the condition called hemolytic disease of the fetus and newborn, which used to be a devastating, all too common tragedy. Modern medicine solved most of this problem with an extremely simple idea: trick the immune system before it reacts. Around the 28th week of pregnancy, and again shortly after birth, if the baby turns out to be Rh positive, Rh negative mothers are given an injection of Rh immunoglobulin, often called Rho-D immune globulin. These are ready-made antibodies that quietly mop up any stray Rh positive fetal cells before the parent's own immune system even notices them. Without that exposure, the body never mounts its own strong response. The immune memory is never formed. The dangerous antibodies are never made. This intervention, small and almost invisible, transformed outcomes. What once caused repeated losses in families now usually results in healthy pregnancies and healthy babies. In many countries, Rh screening is automatic. The test is done early, the injections are scheduled if needed, and the whole drama of incompatibility is reduced to a line in a medical chart and a routine appointment. This is one kind of lasting power Rh negative blood has today. It shapes real decisions in real clinics, quietly steering care plans so that parents and babies can go home together. But that is not why people make long videos and elaborate theories about it. For that, we have to turn to the charged side, the way Rh negative has become part of identity. In the 21st century, many people feel unmoored from older structures of belonging. Fewer live in the same town as their grandparents. Fewer grow up surrounded by a single unified tradition. Lives are mobile, jobs are temporary, online spaces mix strangers who would never meet in person. In that shifting landscape, small anchors can feel huge. A personality type, a horoscope sign, a neurodivergent label, a rare medical condition, or a blood type that only 10–15% of people share. If you have grown up feeling slightly out of step, quieter than your peers, more sensitive, less interested in what everyone else seems to like, and then you discover as an adult, I'm root negative and that's rare, it can feel like a missing puzzle piece clicking into place. Suddenly, there are forums of people who say, me too. I'm root negative, and I've always felt different. Stories start to braid together. I get more static shocks. I have trouble sleeping. I'm drawn to ancient history. I feel other people's emotions very strongly. It is human to see yourself in those lists, to pick out the parts that match and let the mismatches fade. Over time, the group story can become more than the sum of its parts. It becomes a narrative identity. We, the Rh negative ones, are like this. The comfort here is deep. It says, your difference is not an accident or a flaw. It connects you to something, a lineage, a pattern, maybe even a purpose. That is powerful medicine in its own right. Not biological, but psychological. You can see why people defend it so fiercely. If a story has been the first thing that made your loneliness make sense, it is hard to let someone take it apart with journal articles and population charts. The line between comfort and claim, though, is where ethics enters the room. Because these identity narratives can slide almost without anyone noticing from we are different to we are better. The same mechanism that makes group belonging feel protective can, under different conditions, make it exclusionary. We are more empathetic can become we are more evolved. We see through the lies can become we are the awakened few. We have a special task can become we are a higher bloodline. History gives us a long, painful catalogue of what happens when blood is used as a marker of worth. From noble lineages and pure castes to pseudo-scientific racism and eugenic programs, claims about special blood have again and again been used to justify hierarchy, harm, and dehumanization. Our arch-negative mythology lives in a much smaller, more fringe ecosystem than those grand, destructive ideologies. But it brushes up against some of the same patterns. When someone says, half joking and half serious, we're not like the rest of them, we're from another line. They are playing with a fire that has burned many times before. This is why a little ethical caution matters. It is entirely possible to say, my Rh negative status makes me feel connected to a story. I found community in that. Without saying, my Rh negative status makes me superior, chosen, or fundamentally different in kind from other humans. It is possible to cherish a rare trait without turning it into a ladder. From a scientific perspective, the power of Rh negative today is precise and limited. It has practical power in medicine, guiding transfusion choices, shaping pregnancy care, helping blood banks plan their supplies. It has statistical power in research, offering another variable to track. When scientists look at how different traits correlate with health outcomes, disease risks, or responses to treatments. So far, those research lines have found a few modest associations, slightly different risks for certain conditions, but nothing that points to a sweeping, glowing, all-purpose superpower. From a symbolic perspective, though, the power is real in a different way. It is the power of story. The power of a small fact about your body becoming a doorway into questions about ancestry, belonging, and meaning. The power of lying awake at night, scrolling through accounts of other Rh negative people, and thinking, maybe I'm not as alone as I thought. The title of this video asks whether Rh negative blood carries a secret superpower that lasts till today. If we answer in the narrow biological sense, the honest reply is no secret and no superpower. Rh negative is a variation in a blood group system that we understand fairly well. It comes with some specific medical vulnerabilities, which we have learnt to manage. It is not a ticket to psychic gifts, alien origins, or ancient royalty. If we answer in the broader human sense, the reply softens. Rh negative has lasting power as a thread in our shared story. It links a 20th century medical breakthrough to 21st century online communities. It reminds us that a single protein on a red blood cell can mean the difference between life and death in a delivery room, and that knowing about it can turn tragedy into routine safety. It shows how a quirk of genetics can become a mirror in which people search for who they are. In that sense, the superpower is not in the blood itself. It is in our capacity to turn even the smallest facts about our bodies into myths, meanings, and movements. As you drift toward rest, you don't have to choose between wonder and truth. You can hold both. You can imagine the long journey of this trait, from ancient mutations and quiet migrations, through microscopes and maternity wards, into message boards and late-night conversations. You can let the mystery be the scale of the story, not the absence of facts. There is still so much we don't know about why some traits persist in some populations and not others. How subtle selection pressures shape our genomes over millennia. What hidden histories lie folded in the DNA of small mountain valleys and forgotten ports. But we do know this much. Rh negative blood is human blood. It rose within our species. It travels through ordinary veins. It sustains ordinary hearts. Whatever else we build around it, in legend or in longing, rests on that simple shared foundation. In the final section, we'll gather these threads together, the science and the stories, the bottlenecks and the bedside tests, and let them settle into a quieter understanding of what it means to carry this particular variation in a world full of others. So now we're here, at the end of the trail we've been following through science, rumour and memory. If you trace the path backward, the story of Rh negative blood, stripped of its costumes, is surprisingly simple. It is the absence of one particular protein, the D antigen, from the surface of red blood cells. It was uncovered not in a lost temple or a hidden scroll, but in the mid-20th century by researchers puzzled by two linked problems: why some transfusions killed patients, and why some newborns died of a mysterious, relentless anemia. By comparing blood from humans and from rhesus monkeys, by watching which samples clumped and which stayed smooth, they realized there was another layer to blood groups beyond A, B, AB, and O. They named that layer the Rh system, and the presence or absence of the D antigen became Rh positive and Rh negative. From that point on, the story looks like thousands of hospital corridors and quiet labs. Technicians spinning tubes in centrifuges, charts noting a small minus sign beside a blood type, obstetricians ordering Rh tests early in pregnancy, and vials of Rh immunoglobulin quietly preventing a parent's immune system from turning against a future child. In this grounded version, Rh negative mattered because it turned chaos into prediction. It helped transfusion medicine move from gamble to calculation. It turned a recurring tragedy, hemolytic disease of the newborn, into a preventable condition. It gave us one more lens through which to watch inheritance move from parent to child, generation after generation. That is the sober arc, but along the way another story grew, curling around the first like a vine. Myths formed for reasons that are themselves very human. Rarity is one of them. If you learn that only a small slice of the world shares your blood type, it is easy to feel singled out. Add geography, those maps where Rh negative clusters more thickly in some regions than others, and the imagination starts sketching in the gaps. Why here and not there? Who were we long ago that they were not? Layer in identity, the private, sometimes painful sense of being different, of not quite matching the people around you. Give that feeling a biological hook, this antigen you lack, this trait that is uncommon, and it can harden into a story. I am this way because of my blood. And finally add the allure of hidden origins. We are, as a species, irresistibly drawn to secret lineages, lost islands, forgotten civilizations, watchers from the stars. Any trait that looks a little odd on a map, or that shows up at higher rates in a group with a distinctive culture, becomes raw material for those tales. Our arc negative happens to sit at the crossroads of all those forces, statistically rare but not vanishing, geographically patterned but not exclusive, medically important yet invisible day to day. It is almost guaranteed in that position to become a magnet for meaning. When we look closely, some claims collapse on contact with evidence. There is no good genetic support for Rh negative being an entirely separate lineage outside our species, no trace that it marks the descendants of non-human visitors or survivors of a single isolated superior civilization. Most of the tools we use to understand Rh negative population genetics, archaeological context, medical trials, point instead to small cumulative histories. Mutations that arose somewhere in our shared past, drift and selection nudging frequencies up or down in scattered groups. Local advantages that may have existed for a while, then faded, leaving behind only the statistical footprint we can see today. But there are places where even now the line between we know and we're not sure yet stays soft. We don't fully understand why certain regions have such high Rh negative frequencies compared to others. We don't know exactly which pathogens or environmental pressures might have nudged those patterns into place, or how much was simple chance playing out over millennia. We can't yet reconstruct all the tiny decisions who married whom, who migrated, who stayed, that turned a mutation in a few individuals into a feature of whole communities. What we can do, honestly, is map the edges of our knowledge. Within those edges, the biology of Rh negative looks ordinary. A variation among many meaningful in specific medical contexts, modestly interesting in population studies, but not a magic key. Outside those edges there is room for speculation, but not for certainty. And that is where a quiet choice presents itself. You can let the unknowns be an excuse to believe anything at all, no matter how disconnected from what we do know. Or you can let them be an invitation to a softer kind of wonder. In that softer space, meaning doesn't have to be encoded in your cells to be real. If learning you are Rh negative helped you feel less alone, that experience matters. If you found community in others who share that trait, those friendships and conversations are part of your story. If the idea of a rare blood type sent you searching through history, reading about migrations and ancient languages and old hospitals at 3 a.m, that curiosity is its own gift. None of that requires a secret power in the blood itself. It only requires that humans are good at turning facts into stories, and stories into significance. Biology gives you the fact, a missing antigen. Culture does the rest. It decides whether that minus sign is just a lab note or a symbol you wear in your mind. As you lie there, maybe in the half dark, listening to these words while the room quiets around you, you're holding more than one history at once. The deep genetic history in which Rh negative arose among our ancestors and drifted through time. The medical history in which careful observation and experiment turned that quirk into life-saving knowledge. And your own personal history, the moment you first learned your type, the feelings you attached to it, the way it has or hasn't shaped the way you see yourself. All of those histories are real, even if they operate on different levels. The mystery that remains is not a conspiracy locked in a blood test. It is the larger mystery of how small differences intertwine with vast shared patterns. How a protein on a cell membrane can matter desperately in a single pregnancy, and barely at all in the shape of a life. How one variation among thousands can become a global story, because people needed a language for their sense of difference. You don't have to strip away that meaning to respect the science. You only have to remember that meaning is something we make. It hangs in the air between us, in conversations and communities, not in an invisible glow inside certain veins. As this story settles, if you feel like sharing, you're welcome to leave a comment. Say where you're listening from and what time it is there right now. Midnight in a small apartment, early morning on a long commute, afternoon in a quiet room where you're trying to rest. Those small details are another kind of map, another way of seeing how one story ripples out across many lives. If this slow walk through history and myth has helped you relax, or given you something gentle to think about, you can support the channel in simple ways. You can like this video so the algorithm knows it was worth your time. You can subscribe if you'd like more long, calm explorations of the past and the stories we build around it. You can leave a comment with your own experiences of learning your blood type or the stories you've heard about it. All of that helps this little community find each other, the way scattered groups have always done through shared signals, shared questions, shared quiet. For tonight, or for this moment, it's enough to know this. Whatever your blood type, you are part of the same long human line. The same fragile, resilient strand that made it through bottlenecks and plagues and errors and breakthroughs to arrive here, breathing, listening, thinking. Our arch negative is one small variation in that vast tapestry. The real lasting power belongs to the whole pattern we weave together.